third keynote address under the topic of precision phenotyping for crop improvement, one of the key components for the new breeding programs. And then for this keynote, I will invite Dr. Michel Ghanem from CIRAD to join us to the podium, please. Just a small introduction. So Prof. Dr. Michel Edmond Ghanem is a crop physiologist and agronomist working as a senior scientist at the AGAP Institute at CIRAD, the French Agriculture Research and Cooperation Organization. He will be very soon starting, I don't like to say the date, but posted in Senegal in 2024, representing CIRAD as usual. And Prof. Ghanem is the founding chair of the Forgotten Crops Society and the vice president of the African Crops Alliance. He holds an agronomy degree and a PhD in plant physiology from the University of Louvain in Belgium and an accreditation to supervise research from the University of Montpellier, France. He started his career as a research fellow at the CEBAS FISIC, Spain. Since then, he served at the International Center for Agriculture Research in the Dry Areas, ICARDA, as a CGR center. In Rabat, Morocco, as a crop physiologist, where he established and led the crop physiology laboratory that was serving the global breeding and crop improvement efforts of ICARDA mandate crops. Prof. Ghanem was also the program leader for genetic resources at the Land Resource Division of the Pacific Community, SPC, where he led the Center for Pacific Crops and Trees. He was also professor of plant science at the University Mohammed VI Polytechnic, UM6P, Morocco. His area of expertise covers genetic resources, crop modeling, and crop physiology. So let's welcome Prof. Ghanem for this presentation. Thank you. So, Prof. Ghanem, you have around 30 minutes presentation and talk, and then we flow with a few questions from the audience. Thank you.
Thank you, Mais. Good morning, everyone. Happy to be here today to give you a few perspectives from an African perspective on crop phenotyping, exploring boundaries and horizons. And I'll try, it seems that it's not working. No, it's not. Okay. So I'll start by giving a few perspectives on plant phenotyping in the global north. And then I'll give my thoughts about the challenges and limitations of phenotyping in Africa and some perspectives that I would like to share with you. So to start with, plant phenotyping in the global north, as you certainly know, a few definitions. When we speak about phenotyping, we speak about a scientific tool, a discipline to study plant-environment interactions. So the classical G by E that gives the phenotype. It's actually the study of plant structure and function, and it uses non-invasive technology. This is the idea recently. So we can understand how plants really function and how plants cope with their environment. And phenotyping, as Professor Amri said, is used to understand how plants could cope with climate change and different aspects of its actual environment. And it's actually about asking the right questions. It's quantifications of measurable traits, and it's the application of methodologies to measure a specific trait that is related to either a plant structure or a plant function. And it's actually should be driven by something that is question-driven rather than technology-driven. Unfortunately, in the recent years, we've seen the opposite happening. Lots of technological development, and we're quite fortunate to see this happening. But the questions, what do we want to achieve with all this nice technology that you will see examples of, remains intact. And this is where we have to ask the right questions. Also, when it comes to phenotyping platforms, you have various sorts of phenotyping platforms. They go from the organ to even to the cellular part, to aggregated tissues, shoots, root. So they cross different scales. And the idea is how to cross those different scales without losing appropriate information remains actually key in the development of any phenotyping technology. So in the recent years, we have seen also different facilities and kind of activities. Mostly, historically, controlled conditions phenotyping was predominant. And it's actually, the distinction is that it happens mostly in greenhouses, well-defined environmental conditions. And it has been really the scope of the early development of any kind of crop phenotyping. If you can, I'm just showing a few examples of very early great phenotyping platforms. Again, a plant-to-sensor approach means that we bring the plant to the sensor rather than taking the sensor to the plant. And these are two examples that coming from Europe. One is nearby our institute in Montpellier, PhenoArche. It's quite well known, and it constitutes of several types of things. This is maize growing in tubes and being displaced on a conveyor belt. And again, you have NPEC in Wageningen in Netherlands. And those have been developed. If you look at the percentage of the infrastructure, 60% of the current infrastructure are also happening in controlled conditions. And of course, these conditions, like the PhenoArche here, some people here have worked on these phenotyping platforms. They generate lots of information. Just giving you a few examples, you can estimate light interception efficiency on a reconstructed multi-genotype canopy, as you can see here. On the right-hand side, you have the well-watered treatment, and on the left-hand side is a drought stress. But you can see there is a certain variability within the infrastructure itself, which is actually not always completely controlled, as we tend to think of. We can do some reconstruction of so many things. I mean, those are nice videos being generated on sorghum reconstruction. And you can also use it to do some kind of follow-up of tracking of stem tips or even individual leaves in such a nice way. As you can see here in the video that I'm putting, it's a plant that has been conveying to the sensor where it's actually being measured. It also gives you what we call whatever phen extraction. As you can see on these graphs, you can distinguish between the well-watered treatments and the water-stressed ones. It gives you leaf lengths, so many stuff. And this is actually good, except that we are in a position nowadays to ask the question if these robotized indoor phenotyping platforms were really useful so far. And the question is not really easy to answer, but there are two recent publications, one coming from two popes of the phenotyping in Europe, the team of Uli Schur in Germany, and more recently from the team of François Tardieu in Montpellier. And actually, the conclusion is quite not surprising, honestly, because the relationship between the traits that are measured in those highly controlled environments and the yield is extremely dependent on the environmental scenarios, which is absolutely no surprise. And the physiological adaptive traits, unfortunately, so far have not been used or considered per se in a breeding program. I say that being a physiologist, I mean, it's quite painful for me to say that and acknowledge that physiological phenotyping has not really served much, even in the north, contrarily to what we tend to think of. Plus, if you add the fact that most of these phenotyping platforms use pots and, of course, used soil that is absolutely not representative of any real condition in the field. Most of them have constrained volumes, reconstituted soils with peat moss, lots of things that change different types of resources and even how the plants could deal with all of that. So in the recent years, and thanks to the technology development, we have seen a kind of tendency that takes a different kind of approach, that takes the sensor to the plant, and we've seen intensive fields happening, characterization. We also see some kind of lean fields, what we call, those are field sites with very basic equipment to characterize the environment, to monitor what's going on. And in the recent years also we've seen emphasis being put on data and computational services. Of course, modeling has been used for years, and it's quite useful to see that happening. So in the recent years, we've seen platforms like these ones in Arvalis, in Fenofield, on a gantry. So on the left-hand side, you have a picture of a gantry on which you have a series of sensors scanning a certain field. And on the right-hand side, you see a Phenomobile, which is some kind of a robot that goes into the field and characterizes the plant. So those are two examples from the north. What about Africa? As you can see, most of this development has been happening in the north. So what are the challenges and limitations of phenotyping in Africa? And I need to say, before any kind of other statement, that it's a critical component in crop improvement. I mean, there is no secret with the genotyping advances, the bottleneck remains in phenotyping. But it's the case in Africa, but also globally. So don't let anyone fool you saying that the technology developed in the north is absolutely perfect and fantastic, and I'll give you a few examples. So what is the status of the phenotyping platforms in Africa? Let's have a look on this map, and as you can see, most of the phenotyping infrastructure around the world, this is an IPPN survey done 2 years ago, is mostly happening in the global north, and more particularly in Europe. There's a big concentration in France, the UK, and Germany. And if you have a closer look on Africa, very, very few phenotyping platforms do exist. I mean, the map would maybe tell you that there is none, but unfortunately, it's not extremely well developed, and there is a lack of appropriate phenotyping infrastructure, but there are some. So here I have taken 2 examples of infrastructures that do exist in Africa, as far as I know. There might be some others, so it doesn't mean to be extensive and comprehensive. So you have a lysimetric facility with tubes inspired by what ICRISAT has done in Hyderabad in the years. That's in Isra, Bombay in Senegal. And another one in South Africa with a field scan that reminds you of the gantry approach that has been developed elsewhere. So closer to us, I was, during my term at ICARDA, I developed and actually initiated a lysimetric facility that you can see here that later on ICARDA implemented. So that's just here, 100 kilometers from here, and it's developed in Sidi Laidi station. But those are the examples that I am aware of. I'm not aware, and correct me the CGIAR colleagues, I'm not aware of any similar investments being done by IITA, Africa Rice, or countries or centers that operate in Africa. But we have an example just a few meters from here. When I was at UM6P, I initiated, together with my colleague, Professor Moaz Amri, a huge phenotyping platform that is quite a mixture of different approaches, and that you will be able to visit. So that's the famous Phenoma phenotyping platform that you were all invited to visit. So please don't hesitate to contact Professor Amri and send an email to register to visit that platform. So it's a unique, fully automated lysimetric facility that is coupled with imaging in close to field conditions. It has 1,400 mini plots under rainout shelters, and you can measure so many things. As you can see here, that's a lysimetric facilities for measuring water relationship, transpiration in a robot, and also having an automated irrigation system. So it calculates plant transpiration. It could deal with mini plots, and those boxes actually contain soil up to 200 to 300 kilograms of soil. So they are quite representative of what's going on. Still, they are not completely out of the... they also have a coupling with a Phenomobile V2 that was developed by Hyphen in France, and that actually allows you to characterize the shoot system, as you can see in this video. Of course, you can do that at night, but the idea is to do that during the day. It also has controlled phenotyping environments. So again, I'm not fully against the controlled phenotyping environments. I just want to draw your attention to things that are, what do you do with that? So we have developed a long time ago when I was at Icarda this type of machine called the Phenomerzuga that allows you to deal with dual drought, imposing drought on the atmospheric basis and also in the soil. So this is also something that you would be able to visit here when you visit the Phenoma platform. So these platforms are quite nice. So should we have many Phenomas in Africa? Is this the way forward? I personally don't think so. Because of the huge initial cost of investment. I know something about this. You can ask Moaz, who led the project. He knows the huge investment that he and his team has done on this. And we should not forget something, that the initial investment itself is not enough, because you have the follow-up cost on maintenance. And this is one of the major bottlenecks. You could maybe obtain a grant to fund those fancy things, but you need to make sure that they are maintained and they are well kept in the future and they are appropriately used. And this is unfortunately something that is lacking in our continent. So the phenotyping platforms, above the fact that most of them are lacking in terms of infrastructure and the costs are quite expensive, most of the technology has been dealing with northern type of crops. If you look at again, this survey and the recent publication by colleagues even here from UM6P and Rothamsted Research, most of the mentioned species are wheat, Arabidopsis. Well, you might say that maize is an important crop for Africa, but yeah, barley, canola, etc., etc. So where are those crops that are extremely important to Africa? You take pulses, fruits and vegetables, roots and tubers. Most of them are mostly absent from any kind of studies that are conducted. And even when we conduct studies in the global north on crops of interest to Africa, we tend to pick up a few lines that are not representative. Take sorghum. I had a look on sorghum phenotyping, and most of the sorghum that is phenotyped in Phenoarch or somewhere else, they're phenotyped and they're non-tillering sorghum, which is really not always very much representative of the African reality. And African crops, and here you have a list of huge African crops, teff, finger millet, fonio, other plants that are close to my heart that we call forgotten or neglected crops. Where are they? They're not present in the development, and they would need some tweaking so that they are, the technology is able to deal with them. Of course, you might argue that these things are quite general and you can adapt it to any crop, but you still have some tweaks. I mean, it's not really straightforward to phenotype an easy plant with an easy architecture like wheat or maize, and then say, let's go on and extrapolate that on teff or even phonium. Yesterday, I was quite struck by the fact that most of the fields that were presented in publications looked like these ones. They seem to be coming from a nice and clean field in the north, or maybe in Brazil, I don't know. But they're not very representative of the African reality. I mean, most of the fields that you see in Africa do not look like this nice and fancy field that is the kind of expected setup that we do in field conditions. So how do you deal with the spatial and temporal variation in field conditions? Take that example in Niger. I'm sorry because the visibility is not so good. That's a millet field grown in the Sahel, and you can see there is lots of heterogeneity. There's spatial and temporal variation in field conditions. So how do you deal with that kind of variability when you do phenotyping? How do you deal that? That's the effect of human settlement activities. Most fields are also, if we want to open also avenues, new avenues of phenotyping, there is also the agroforestry that has been that has taken lots of importance recently in the European context and in the north globally, where you have intercropping or mixed croppings, trees, and etc. Most of the experimental setup look like this one, which is quite nice. You have very much aligned type of trees, and next to it, nice fields that are in between. And yet, even with those nice-looking fields in the north, agroecology hasn't really, or agroecological approaches haven't really benefited from progresses in phenotyping, even in the north. So don't let anyone fool you and tells you that we have everything set up for the south. The reality, even in agroforestry in the south, is extremely different than what we could see in the north. Look at this ancestral Federbia pearl millet intercropping system. That's another example of a project in Sustain Sahel led by CIRAD. And those agroforestry parkland are actually part of the West African smallholder agricultural landscape. And there is a high spatial heterogeneity of the millet crop that is being growing under those, finally, those trees. So that's an experimental setup, and it doesn't really look like the one that we are quite used on seeing in the north with nicely aligned, very homogeneous type of trees. It's not representative, again, of the African reality. So how do you deal with that? How do you deal with that random intercropping? And that, again, the actual setup in the northern way of thinking is quite aligned, trees. So it's easy to fly drones and characterize that. While the actual implementation is far from the standards of what we might think as the standards. The actual implementation of those trees is extremely different. And that's an example, and I here invite you to listen to the presentation of my colleague, Romain Fernandez, about the effect of Federbia. So he had to look at the effect of Federbia on millet growth in different places, and the influence of all of that, and he had to call upon approaches that has been used in human medicine. So like the probabilistic, what he calls the probabilistic atlas of Federbia, just to implement the effect of Federbia on the surrounding things. And that's another example about the control that Federbia exerts on its surrounding. So you could also characterize what's happening in the millets field. Remember, so he had to develop another set of approaches to look at the F-cover around that Faidherbia and to see where is the effect of that crown stops and where it goes beyond that. So that's spatial heterogeneity. We talked about different systems, even in agroforestry, but you also have to deal with different types of production systems. So you have different systems. If you just take an example of sorghum, sorghum is cultivated under either soil cropping or rotation. It's also being intercropped with cowpea. You have intercropping with groundnut. So how do you deal with that variability? I mentioned the example of parkland agroforestry, which is another way of dealing with a production system. You also have different sets of these things, half moon, post-rainy, etc., etc. So the classical G by E by M, you add on top of it a G by G by E by M, and then you add an S. You have a wide array of production systems that really don't quite look like the ones we are used on considering in the north. So again, you need to deal with that, and deal with heterogeneity is an important thing to be developed if we want to implement some of that technology in Africa, because the African landscapes are extremely not comparable to those that exist in the north. So if you don't take that into consideration and you just import a random technology and use it as it is, it won't work, unfortunately. So there is another problem. That's an example from, again, from a colleague of mine, Dr. Cornet at CIRAD. He wanted to have a multispectral sensor and use it in tropical conditions. So he realized that there were some issues with the radiance values and that some constants that were fixed in the machine itself were based on conditions that were developed in temperate regions. So if you take that to tropical regions, most of the radiance estimates are wrong, and they're actually not reflective of what the reality is. And you will have extremely, errors in estimating vegetation index and so on and so forth. That's an example from 2018. The manufacturer was aware of that, and I hope he fixed the problem. But again, that's a problem that one might face when you work in conditions different from which your machine or equipment has been initially developed. So the material is most of the time unsuitable, and you need to do some tweaks in order to make it useful for everyone. There is also the problem of data infrastructure. Just to give you an example, these platforms, nice and good-looking platforms, generate huge amount of data sets. Just an example of what's going on in Feno-Arche, and you can see here, if I take two sides, one top view, you have around 30 terabytes of data per year. And if you add that to a multiple network of phenotyping platforms, you can imagine the huge data sets that are generated in that. So if you don't take that into consideration and you don't develop a data infrastructure, I took an example of here a super calculator in Senegal. Unfortunately, it has been bought, which is good. But as far as I know, since January 2022, the calculator has not been working. The data center is not fully operative, so you can't use it. At UM6P, we're quite lucky to have the data center, one of the biggest in Africa, if not the biggest, just across the street. So Moise and his team will be benefiting from that when he will make Phenoma work. So again, the data infrastructure is nice, but you'll also have to consider that this is also a scientific field. It's not that I can just call upon some of the colleagues from the data and tell them, I have some images, just read them for me. It's a science per se, and it really needs a workflow. It's quite data intensive, and it needs to be treated as such because it needs to be treated as a distinct scientific discipline. So it's not always enough to invest in data infrastructure by itself. You also need to invest in the science of the data next to it. And for that, you also need to invest on technical skills and personnel. Unfortunately, most of the talented and skilled personnel in Africa that are trained are either working in the north or for some reason they have been working somewhere else. And even when you train them abroad, the training is really unsuited sometimes to local demands. And I go back to what was mentioned yesterday in developing African talents for Africa and in Africa that are suitable for the condition itself. You also have a problem. I'm sorry for being pessimistic this morning and letting you bombarding you with a full list of big issues. You have also problems with regulations that are controlling the emerging technologies. I tried to fly drones here in Morocco, and it was quite a bit challenging thing to do because you have lots of regulation, and I'm not saying that you should not need regulation. They're here for a reason, but you have a series of authorization to acquire from the Air Force, from aviation and police, et cetera, just to undertake a drone flight. So it makes it unequal for certain countries to deal with those new technologies like UAVs. If you want to do that in other places, in other places might be easy. I mean, my colleague Alain Audebert has an extensive experience in running flights in Senegal, and I think it's easier than any other places. So this makes that some of the technology can only be implemented by people who have time and who have means to do that. And it's unfortunately not the case in most of the African institutes. Another problem is the weak linkages and the weak inter-African collaboration. I mentioned the work of Alain in West Africa, developing field phenotyping network in the frame of the DPRO. Unfortunately, this is not a generalized example, and so far, we don't have an African plant phenotyping network. We're trying to fix that with some colleagues from the IPPN, MICE, and others, but we don't have that so far. So you don't have the you don't have the inter-African collaboration. You don't have big projects like Emphasis that is implemented across Europe that allows people to share things and look at that. And we're contemplating recently the launching of a project that deals with networking more than anything else. So now what? After all this list of depressing problems, it's actually what I try to say, that the reality in Africa is not the same as in the global north, and that most of the developments have been made in the global north. And if we want to take that technology to the south and we want to implement it, there is a series of things that those who develop and implement need to deal with. So I think there are a few ways that I would suggest here. First, public-private partnership. I mean, this is something not new, but this needs to be encouraged, specifically in the field of phenotyping. Most of the donors don't really like to consider projects that are addressing just plant phenotyping per se. You also have the problem of the technology itself that allows you, the cost of the technology itself that allows you to deal with something that has a good throughput and has a good quantity of the data. The investment on the scientific skills. Again, developing of pipelines that are exclusively and initially developed for the African crops instead of just growing the same good old species. And most importantly, we need to get affordable, easy to handle, reliable phenotyping tools that could address the problem. So I'll finish with that. I just want to say the following messages. There are some challenges, but there are also ways of getting out of it. Having been involved in the implementation of some of the phenotyping platforms in the South, I invite you not to be fooled, I dare the word, by the technology itself. Ask the right questions, see if those questions could be addressed with an easy and a low-cost phenotyping device. and above all, improve the quality and of the data, the way that you deal with the data, get the interaction between the different disciplines. You need agronomists, physiologists, breeders, and data scientists to come together to solve that thing. And of course, increase the implementation of inter-African collaboration in that field. With that, I do thank you.
Thank you, Michel. It is really a very nice presentation going through all these phenotyping platform development and what could these platforms or these new technologies bring to accelerating the genetic gain and somehow speed up having first a good quality phenotyping data and speeding up the breeding programs. Interesting to see that you are the way that you are tackling things and going from taking the plant to the sensors, and now the approach is, and it would be better to bring the sensors to the plant, and this takes out somehow what we develop indoor, especially when it comes to phenotypal platform. And for indoor platforms, when you develop the technology indoor, there is big challenge to bring it to the farmer field. And it would be really interesting to bring this technology outside to the field, so under what we say close to field conditions. The Phenoma platform that we have, and then there was some videos, it is close to field platform. That means the only thing that we can control is precipitation, that means rain, but all the other conditions are close to field conditions. The second thing, when we designed the platform, Phenoma platform, it was somehow a debate between a physiologist and a geneticist. And then usually for the phenotyping platform, there is lysometric system for single plant phenotyping or testing. We said one single plant don't represent one genotype. We need one population of plants representing one genotype. This is why we choose to go for mini plots, that means 60 by 60 by 60 centimeters, to have a population of plants where we can respect the same density and planting protocols. This was a command. Maybe another interaction when, before we move to the question from the audience, is G by A by G by G by E by M. I think we can consider the second G as part of the E. And this is another approach. Maybe other agronomists, they can comment on that. And for the network, just to say, and then my colleague Enoch here from Benin, we got approved for funding one project, EU-funded project, for somehow reinforcing the network between African institutions, and UM6P is part with other institutions from Africa, and this is to use the high throughput phenotyping technology that we have at UM6P, but with other countries from Sub-Saharan Africa. So with this, I will maybe ask for a few questions from the audience. We can start with a few here, and who has questions from the audience, please? So we can take some questions. So Moha, is there any? Yeah, please. Dr. Moha from INRA. Yeah. Yeah. No. Your hand.
Non.
Yeah.
Derrière, je suis pas devant.
Ouais.
Il y a quelqu'un derrière. Premier rang, premier rang.
Thank you. First of all, I want to thank Professor Ghan for his frank presentation and the expected output from this phenotyping question. I will be also frank in my probably what we see in phenotyping, the investment, as you said, it's a big investment, and one institution could not afford that investment. And my question is the output worth it to have those such investment, the output from those phenotyping platform, or just it's good for publications and having papers coming out? That's my
yeah.
Thank you.
So thank you for your frank answer. I'll get for your frank question. I'll give you an honest question. If people like François Tardieu felt the need after 30 years of their career to publish a paper that has been released maybe 2 weeks ago, justifying or saying, yes, it is useful to do all those fancy things in the controlled environments, I think that the answer to your question is yes. I mean, it was useful to some extent. I'm not advocating against the controlled environment type of phenotyping and huge investment. They're useful for people like me who want to decipher some of the traits and to look into something that is extremely small. But honestly speaking, most of the development of the infrastructure indoors did not really benefit a lot to the breeding pipeline. And it's not only in Africa, even in Europe. I mean, it didn't do that much. The need for justification papers after such a long careers, it tells you a lot. And I think we have colleagues that work in those fields that know what we're talking about. And I say it, I mean, I'm a physiologist. I spent lots of time at ICARDA and other places developing those platforms. And I really want to say, let's go to the field. You know, let's not just stay in controlled environments.
Thank you. So we have another question from Omar Idrisi, please. Yeah. Another mic here. Okay. So
Yeah,
let's do with Michael Alberton and then move to Omar. A quick question, please, and quick answer.
Yeah, so Michel, I'm just really following up on that, and you're talking about the lack of success, if you like, of physiological. So what do we learn from that? And what's the way forward if we're still interested in relating physiology to breeding? How should we go about it?
Okay, so unfortunately, physiologists and breeders and agronomists used to work each one in their caves. And, you know, physiologists would go out of the cave and say, hey, we've got a trait for you, and go back to their cave. And that's why those things were not taken. I think there is no recipe here except then integration, having people talking to each other from the very beginning, confronting the different approaches. You know, we don't have necessarily the same type of approaching things. Physiologists, breeders, agronomists, etc. Confronting that together, I think this is the key, and it hasn't happened so far, unfortunately.
Thank you. Omar, please.
Yes. My name is Omar Idrissi. I'm from Inra, Morocco. Thank you, Michel Ghani, for this interesting talk. I have a question about this African plant network phenotyping. I have the opportunity to participate, let's say, to the international plant phenotyping network. It was a quite successful initiative, I think. Many people from around the world comes to this what you call the North facilities and North phenotyping platforms. Could you please elaborate more about this initiative that you have said about African Plant Network? What would be the involvement of, I think, the CGR centers? I think in Africa we have, to my understanding or to my knowledge, 3 or 4 international centers, CIMIT, ERI, ICARDA, and things. Maybe it could be a good initiative, and maybe here, I don't know the state of the things for the project, if it could be initiative targeting practical breeding for the real impact in Africa, let's say. Thank you.
Okay, thank you, Omar. The idea is actually an answer to a European call about infrastructure that Sirad and colleagues from INRAE and other colleagues also from the IPPN and Moise and his team in UM6P, but also others are championing, is the fact of let's try to increase that interconnection between the African science community around phenotyping and with the European science community on phenotyping, and see how we could benefit from that exchange. As I said, not importing any ready-made solutions that were made up north, but rather adapting them to the conditions of Africa.
Yeah, last question there, please. The last one, and then we move because we have another. Yeah, please.
Yeah, thanks, Michelle. Yesterday we heard the concluding remarks from Dr. Rodomiro Ortiz, who said that 40 years back, we were going to the field and doing the phenotyping ourselves, and now it is going into more and more imaging or precision phenotyping, doing all of that. And we also know that there is a lot of QTLs that has been identified for the quantitative traits, which actually has never been used in actual breeding except the publications. And we are now talking about precision phenotyping, getting into all these images, and then using algorithms to get solutions to it. I want to just understand again, as a kind of mid-age plant breeder or molecular breeder, kind of where I should be, this side or that side, and how good this precision will be when I deal with quantitative traits. Thank you.
So quantitative traits and what I call integrative traits, say like yield or any kind of things, there should not be they're not a trait for me in the sense of a phenotyping objective. The answer to your question would be you should be on both sides, probably, but with the appropriate questions. The problem that I have seen throughout the years is that sometimes we are becoming technology-driven. We can do things because, lots of things, and luckily we're quite fortunate to do that, to have that opportunity in doing this. But where are the questions? It's like having all the answers, but where are the questions? Ask the right question, say what kind of tool would answer your question, and the tool might not be really an expensive machine that costs $2 million. It could be a very easy type of device. It could be a smartphone. It could be so many things that are quite affordable. So I think we should be on both sides. We should benefit from all that development, but with the right answers and the right questions, more importantly.
With this, thank you, Michelle. Thank you for all. Just before I leave the floor to my colleague Aijamang and the other keynote speech, Jill, two announcements, please. For those who are interested to visit the platform on Thursday, please register. The deadline is today noon. So if you are interested, please do it before noon today. For logistic arrangements, then when we bring transfer participants from Marrakesh to here, so we need to prepare all the logistics. The second one, for those who have payment on spot, there is someone who is available on the registration desk, so please proceed with the payment for the APBA registration. I'm talking about the registration to the conference. Thank you very much, and then welcome, Ajman.
Thank you, Moise. Good morning, ladies and gentlemen. So we have another interesting presentation coming up, which will be the fourth keynote address on the topic, Achieving Sustainable Productivity in Agriculture Through Beneficial Microbial Associations. And this presentation will be given by Professor Giles Oldroyd. Professor Giles Oldroyd studies the mechanism by which plants form beneficial associations with microorganisms, both bacteria and fungi, that aid in the uptake of nutrients from the environment, including nitrogen. The long-term aim of his research is to reduce agricultural reliance on inorganic fertilizers, and he currently leads an international program funded by the Bill and Melinda Gates Agricultural Innovations. He completed his PhD in 1998 at the University of California, Berkeley, studying plant pathogen interactions, and then moved to Stanford University to work on nitrogen fixation in the laboratory of Professor Sharon Long. After working 15 years as a group leader at the John Innes Center in the UK, he moved to the University of Cambridge in 2017. And in 2019 was elected the Russell R. Geiger Professor of Crop Science. In his role, he directs the Crop Science Center, an alliance between the University of Cambridge and NIAP. The Crop Science Center strives to deliver transformative technologies that sustainably increase agricultural productivity for all the world's farmers. In 2020, he was elected a fellow of the Royal Society and a member of EMBO. And then in 2021, he was elected foreign member of the National Academy of Sciences in the US. With a round of applause, please help me welcome Professor Giles Odroyd to the stage. You would have 45 minutes in total.
Yes. Yeah. I hope I leave plenty of time for questions. Okay. Can everybody hear me, or do I have to bend down here? Okay. Great. Thanks very much, and thanks to the organizers. It's my first time at the African Plant Breeders Association. It's wonderful to meet a lot of new people. So our agricultural systems, at least in the global north, are highly dependent on the use of inorganic fertilizers. This is the green revolution. And while it's massively driven up productivity, it's a rather inefficient system. A lot of the nutrients we apply escape into the environment, where they cause severe biodiversity loss, both in aquatic and in terrestrial ecosystems. And we also lose a lot of the nitrogen that we apply to the soil in the process of denitrification that causes volatilization of nitrogen to nitric and nitrous oxides. These are causes of air pollution and also potent greenhouse gases. So while we've got a great system for driving up productivity, it's a very inefficient system that causes significant agricultural environmental problems from our agricultural systems. For a high input farming system, it's really the access to nitrogen is driving an unsustainable practices. But the opposite is true for a lot of smallholder farmers, particularly here in sub-Saharan Africa, who really lack access to these inorganic fertilizers. Seem to be interesting. Seem to have missed a couple of slides, but that's fine. So the most smallholder farmers are really not using large amounts of inorganic fertilizers. They have very little access. In some cases, they do have access. A lot of farmers do have some access, but it's certainly not enough to drive up those productivity gains we see in the global north. But it is this access to inorganic fertilizers that limits a lot of their smallholder crop product production, in particular for their cereal crops. So I am working on a program that's striving to drive innovations in this area and reduce the dependence on inorganic fertilizers. And I propose, and this is entirely my proposition. I'm sorry, my slides have got a little messed up in the transfer. But I propose that there is an opportunity probably to, maybe it's optimistic, but to eradicate the need for inorganic fertilizers by about 2050. And I see that there's about 3 different phases. There's not a single silver bullet that's going to do that, at least not in the short term. So what's happening a lot right now, and I apologize for the messed up logos on the slide there, but there's a lot of companies, particularly in the USA, that are investing in inoculants. These are associative bacteria that colonize the roots, the rhizosphere, they colonize the intercellular spaces, and they have the ability to fix nitrogen within the free-living state. Now, this is a big investment, and I've no doubt that this is going to certainly reduce some of the nitrogen that we have to apply through inorganic fertilizers. However, these free-living nitrogen fixers, they have to be super competitive in the environment, they have to replicate to very high concentrations, and yet they have an intrinsic disadvantage in that they're fixing nitrogen and exuding that nitrogen to the plant. So in my opinion, that we're really going to reach a ceiling with how much the plant's nitrogen you can deliver through this route of inoculants, and I would estimate that's in the range of 25 to 30% of the plant's nitrogen can be delivered through these associative free-living nitrogen fixers present on the plant surface or in the intercellular spaces. And there is another issue with these inoculants, getting inoculants to smallholder farmers. So some of the inoculants that are currently available, they have to be refrigerated for transport. They have to be applied on the field very quickly after they've been delivered. And this is obviously rather challenging in a smallholder farming system that lacks that infrastructure for refrigeration throughout the process. I would say the second phase, which is enhanced nutrient use efficiency, there's many routes to this: structure of the root system, the efficiency with which the plants use nutrients, and then also associations with microorganisms such as arbuscular mycorrhizal fungi can increase the efficiency of capture of nutrients from the environment and are certainly going to help in reducing our dependence on nitrogen fertilizers. But if we want to completely transform whether our use of our access to nitrogen fertilizers, then you need a truly transformative technologies. And I would say that that's a sort of the phase 3 of this process, which are the more transformational approaches, such as either putting the nitrogenase enzyme into crop plants, engineering it in the mitochondria, or transferring the symbiosis that legumes perform with nitrogen-fixing bacteria and transferring that to other crop species. And in my talk today, I'm going to highlight 2 aspects that we're working on. An aspect of phase 2, that's work with arbuscular mycorrhizal fungi, and an aspect of phase 3, which is the transfer of the legume symbiosis to other species of crops. So in the ENSA project, we are inspired by legumes. Legumes can really access pretty well all of their nutrients from their environment. They really do not depend on the use of inorganic fertilizers. They have associations with arbuscular mycorrhizal fungi. The colonizer roots ramify out into the soil and really massively expand the root surface area for the uptake of nutrients. But they also can associate with nitrogen-fixing bacteria. Now, I'll just show you first the interaction with mycorrhizal fungi. So this is not limited to legumes. This is quite a widespread association. You can see here some plant roots that are being colonized by the fungus. You can see the fungal hyphae ramifying out into the soil, making spores. And once they colonize the fungus, on the top right-hand corner there, you can see the fungal hyphae invading the cortical cells of the plant root and creating these highly branched structures, which are areas for nutrient exchange. The plant is delivering the fungus with all of its carbon needs. And in exchange, the fungus is ramifying out into the soil. It's capturing phosphates, it's capturing nitrates, it's capturing micronutrients, it's also capturing water. So these fungi, although they're very well characterized for uptake of phosphates, are actually delivering many of the plant's nutrient needs. And you should really think of them as a massive expansion of the potential surface area of the root and its ability to colonize the soil. Legumes also have this association with nitrogen-fixing bacteria, and that allows them to access what is really an unlimited and very sustainable source of nitrogen in the atmosphere. 70% of the air we're breathing is nitrogen, but it's only bacteria that can access that nitrogen through the enzyme nitrogenase that converts it into ammonia, form of reactive nitrogen that plants can use. So legumes are really a fantastic vision for where our crop plants could be in acquiring pretty well all of their nutritional needs through these beneficial microorganisms. Now, nitrogen-fixing bacteria, the association with nitrogen-fixing bacteria leads to the production of these structures, these nodules. They're very specialized structures on the roots of the plant. These are these pink structures that are coming off these nodules. And if you take a transection through those nodules, you can see the right-hand cell there is a cell that's infected with bacteria. You see many bacteria colonizing that cell, surrounded by plant-derived membranes. And in the middle there, you can see that compares to an uncolonized cells. So in both these symbioses, both mycorrhizal and the nitrogen-fixing symbioses, these lead to intracellular colonization of the plant cell by these microorganisms, massive intracellular intrusion of these plant cells. And that's very different to most other plant microbe interactions. Most other plant microbe interactions, the microorganism is predominantly colonizing either the surface of the plant or the intercellular spaces, but very rarely showing such extensive intracellular colonization. So our vision for the ENSA project is essentially demonstrated here. So can we maximize the use of pre-existing arbuscular mycorrhizal associations? And can we expand the microbial associations with which the plant engages to also include the interaction with nitrogen-fixing bacteria? Essentially converting our cereal crops much more to something analogous to what we see in legumes. Now, in the current situation, our cereal crops have the ability to associate with arbuscular mycorrhizal fungi. But the association with mycorrhizal fungi, while it's very common, most of our crop plants can engage with mycorrhizal fungi. It's really not been a target for plant breeding at all. I'm not aware of a single plant intentional genetic improvement of the arbuscular mycorrhizal association. In fact, you could argue that in many ways traditional plant breeding approaches have actually restricted the utility or selected against the mycorrhizal association. Much of plant breeding, at least in the global north, is in high nutrient environments when the plant does not choose to engage with the fungus. And so you're breeding your crops in a context where the fungus is mostly absent and its services are not present in your breeding systems. So there is an opportunity also to intentionally enhance the association with arbuscular mycorrhizal fungi and almost domesticate that trait in order to see its greater utility and greater usefulness in agriculture. Now, already, just the existing mycorrhizal fungal associations that have really not been intentionally genetically improved, they still can account for quite a significant amount of the crop yield. So this is a study that my colleague Rory undertook in maize mutants. So on the AMF resistant is a mutant of maize that is not able to associate with arbuscular mycorrhizal fungi. The AMFS is a susceptible, that's your wild type plant that can be colonized by mycorrhizal fungi. And this is in a medium input subtropical field context in Mexico. And you can see that the AMF-resistant lines are not looking very healthy. They're quite yellow. Their nutrient status is somewhat limited. And in this study, they accounted that the mutant, the AMF mutant, yielded about 30% less than the plant that could be colonized by mycorrhizal fungi. So under certain conditions, mycorrhizal fungi can be supporting quite significant yield benefits in our existing crop plants. Now, the plants, we identified this transcription factor, and I'm really not going to go into a lot of the genetic details today on this work, but we identified this transcription factor called NSP2. You can see here some barley mutants, barley mutant NSP2-1. It's severely reduced in its colonization by mycorrhizal fungi. We're measuring percent root length colonization here, so about 75% of the barley root gets colonized by mycorrhizal fungi. But when we overexpress that transcription factor, we see hypercolonization of the plant by arbuscular mycorrhizal fungi, both under low nutrient conditions, but also under high nutrient conditions, so high phosphate environments, which normally completely inhibit the engagement with the fungus. We now see with the NSP2 overexpressing lines that they still get well colonized by the fungus. So we wanted to ask, this is what we observed in the laboratory situation, can this translate into a field context? And so at the Crop Science Centre at the University of Cambridge, we undertook field trials with these genetically modified barley lines as well as the CRISPR gene-edited lines. And here I'm showing you the results from our first field trials. And you can see that the mutants, the NSP2 mutant doesn't get colonized at all by the fungus in a field context. The wild type is showing about 5% of its root length colonization. And in contrast, the overexpression lines are showing about 30% of root length colonization, almost a tenfold increase in total colonization of mycorrhizal in the field. I want to just highlight something in this that's demonstrated in this slide. In the previous slide, you can see that mycorrhizal colonization, at least in a laboratory context, can get to almost 100% of the root length is colonized by the fungus. But actually, in most field conditions, we see very, very reduced levels of colonization in a normal field context. And this is very low, seeing about 5% of the root length colonized. But this is not unusual, that in fact when we put our crops out in the field, there is a much greater opportunity for them to engage with mycorrhizal fungi. And the genetics that we're discovering, like for instance the NSP transcription factors, are an aspect that control that total level of engagement. Now, currently, these are genetically modified lines that we've used as experimental tools to see if we can see impacts in the field, and that's exactly what we're seeing. But we are now exploring gene editing approaches, which I think will be much easier to deliver in an impact context. But I also expect these transcription factors that are really responding to the environment and controlling how much the plant engages with the mycorrhizal fungi. They're very likely to be targets for selective selection, and probably also within natural diversity could also be useful in a plant breeding context to increase engagement with mycorrhizal fungi. So we have the ability now to massively drive up by about tenfold the plant's engagement with mycorrhizal fungi. And in order to see true impact, what we're currently doing is stacking that with existing genetics within the crop. Something that's not very well appreciated in beneficial microbial associations, certainly with mycorrhizal fungi, is that the amount the plant benefits from engagement with the fungus is genetically encoded. And what I'm showing you here is demonstrated in wheat, but it's equally true, it's been demonstrated also in rice and in maize. And these are different elite wheat lines. And we're measuring the shoot biomass benefit that is accrued from the presence of arbuscular mycorrhizal fungi. And you can see that some wheat lines actually have a penalty from engagement with mycorrhizal fungi. So on the left side, panel A is actually a 30% penalty. So the fungus is actually parasitic on that wheat variety, and it can go all the way to 100% improvement in shoot biomass because of the presence of arbuscular mycorrhizal fungi. And I think that this goes to the core of what I was arguing in the first place. We have not been selectively and intentionally breeding our crop plants for benefit from arbuscular mycorrhizal fungi. And in fact, sitting in all of our elite cereal crops, there is this variation for benefit that is accrued from these beneficial fungi that ranges from them being parasitic all the way to doubling shoot biomass. And so at the moment, we are currently stacking that hypercolonization through engineering with the NSPs into these highly benefiting wheat varieties. And we just have the T0 lines right now for that H variety stacked now with hypercolonization to see if that really boosts productivity very significantly. So I would say that in the next 10 years, or certainly within this decade, I believe we're going to be seeing a big change in the utility of arbuscular mycorrhizal fungi. Certainly, we've got the handle on the genetics and understanding how to drive improvements with arbuscular mycorrhizal fungi. And what's also emerging in this is the inoculant industry that's innovating with regard to how you produce inoculants using mycorrhizal fungi. The much harder aspect and the much more challenging aspect is the right-hand side of this panel, which is transferring a completely new symbiosis into cereals and encouraging cereals to engage with nitrogen-fixing bacteria. Now, arbuscular mycorrhizal associations here, I'm showing you a phylogeny of the entire plant kingdom. When we know that arbuscular mycorrhizal fungal associations evolved about 450 million years ago, right at the very, very earliest onset of early land-plant evolution, the arbuscular mycorrhizal fungal association was a very early innovation in that plant evolution. And this is why the mycorrhizal symbiosis is so ubiquitous with the plant kingdom and why so many of our crop plants engage with beneficial mycorrhizal fungi. In contrast, the nitrogen-fixing symbiosis evolved somewhere between 80 and 100 million years ago, and it's restricted to a group of about 25% of angiosperms that can engage with the nitrogen-fixing symbiosis. However, what we do know is that when the nitrogen-fixing symbiosis evolved about 100 million years ago, it didn't reinvent the wheel. In 20 years, or now even 30 years of genetic studies in legumes on nitrogen fixation, we have not found a single gene that doesn't exist in non-nitrogen fixing plants. So at that point of innovation 100 million years ago, there wasn't new gene innovation. There wasn't innovation in new genes. It's not that legumes have a complete set of new genes that facilitates its engagement with nitrogen-fixing bacteria. What we are finding, and what I'm going to highlight in the second part of my talk, is that what legumes actually did is like a pick and mix. They took this bit of genetics that pre-existed and this bit of genetics that pre-existed and another bit of genetics, re-networked them in a novel way that facilitated the engagement with nitrogen-fixing bacteria. But there is no single set of novel genes. And key to that is this signal transduction pathway, or key demonstration of that is this signal transduction pathway that allows recognition of nitrogen-fixing bacteria by legumes, recognizing the signaling molecules, the so-called NOD factors, or lipoketo-oligosaccharide signaling molecules that nitrogen-fixing bacteria are producing, and that allow legumes to recognize those nitrogen-fixing bacteria. Now, this was a signal transduction pathway that myself and others genetically dissected in model legumes, and each of those proteins, essentially, if you mutate them, you abolish the engagement with nitrogen-fixing bacteria. And we could secondarily stack them into this coherent understanding of how this symbiosis signaling happens. Now, that signal transduction pathway, when we did the genetic dissection in legumes, we discovered it because it was essential for the engagement with nitrogen-fixing bacteria. But if you mutate any member of that signaling pathway, you also abolish the engagement with arbuscular mycorrhizal fungi. So that was it seems like this is a signal transduction pathway that evolved 450 million years ago. So that signal transduction pathway didn't evolve at the 100 million year point with the evolution of nitrogen-fixing associations. It actually evolved at the 450 million year point at the onset of the arbuscular mycorrhizal symbiosis, and then was recruited later on when legumes evolved engagement with nitrogen-fixing bacteria. And we see this, particularly this, again and again in the nitrogen-fixing symbiosis. The nitrogen-fixing symbiosis is a modified form of the engagement with arbuscular mycorrhizal fungi. And that's true for the signaling pathway, but it's also true for how the plant is colonized by these microorganisms. Is using the same genetic components that the plant uses for facilitating fungal colonization as you get for facilitating bacterial colonization. Now, to demonstrate that that's the case, I'm showing you now mutants in barley. These are mutants which are generated by CRISPR in barley that take out multiple components of that symbiosis signaling pathway, and we're quantifying colonization by arbuscular mycorrhizal fungi. And you can see that if we mutate any component of that symbiosis signaling pathway, you completely abolish the engagement with arbuscular mycorrhizal fungi. So it's showing you that that signaling pathway is absolutely present in our cereal crops and is facilitating engagement with arbuscular mycorrhizal fungi. And what's more, you can even take the genes from cereals, transform them into legumes, and they complement legume mutants not only for engagement with the fungus, but also for engagement with nitrogen-fixing bacteria. So the signaling pathway is there and is doing exactly the same job in a cereal as it does in a legume, associating with nitrogen-fixing bacteria. So core to the signal transduction pathway are oscillations in calcium. This is how the signal transduction pathway transmits information from the cell surface into the nucleus, and those calcium oscillations drive gene expression changes. And here I'm showing you symbiotic calcium oscillations, which is a feature of this symbiosis signaling pathway, not in a legume, but actually in barley, on the surface of a barley root, responding to signals from nitrogen-fixing bacteria. This is another way to show you those symbiotic calcium oscillations. On the bottom of the panel, NSLCO, CO4, CO8. These are signaling molecules produced by arbuscular mycorrhizal fungi, and you can see the activate calcium oscillations in barley, these symbiotic calcium oscillations. On the top, SMLCO and MLLCO are actually the signaling molecules for nitrogen-fixing bacteria. SMLCO is Cynorhizobium meliloti. It's a nitrogen-fixing symbiote of Metacargo. ML is Mesorhizobium loti. It's a nitrogen symbiote of Lotus japonicus. And you can see that barley is also able to recognize the signaling molecules even from these nitrogen-fixing bacteria and activate the signal transduction pathway. So the whole signal transduction pathway is present in cereals that allows them to perceive beneficial microorganisms and activate downstream responses. So why is it then cereals don't engage with nitrogen-fixing bacteria? Well, one of the key innovations that we know that did happen in legumes associated with engagement with nitrogen-fixing bacteria was the activation of that signaling pathway and an induction of a developmental change in the root, and that is the production of nodules that allow engagement with nitrogen-fixing bacteria. And there is no such developmental process happens when the plant engages with arbuscular mycorrhizal fungi. Now, if you activate any component of this signaling pathway in a legume, using gain-of-function mutations, and on the right-hand side, the bottom C and D are plants that have been activated by the pathway using a gain-of-function in CCAM-K, the calcium decoder. And if you use a gain-of-function in this pathway in legumes, you get spontaneous activation of nodules in the absence of applying rhizobia or in the absence of applying the signaling molecule. So that tells you in a legume, just turning on the signal transduction pathway is enough to turn on the whole process of making a nodule. Now, if we use those same gain-of-functions in cereals, we see no developmental changes. So it tells us that a key innovation that happened in the nitrogen-fixing symbiosis in legumes was that the signal transduction pathway could activate a developmental change within the root of the plant. So we wanted to really ask, and in the ENSA project, we are as much a discovery project as we are an application project. We have to discover the key innovations that led to this ability for symbiosis signaling to now activate a structure, a nodule, that can accommodate nitrogen-fixing bacteria. And we really wanted to ask, what is the key innovation? What differentiates lateral root, which is the structure on the left, to a nodule, which is a structure on the right? And certainly one of the big key innovations is that nodules accommodate nitrogen-fixing bacteria and get heavily colonized by those bacteria within those cells. So, but how a lateral root forms and how a nodule forms, at least if you look at the very earliest cell divisions, very, very little differences between a lateral root and a nodule. And at these very earliest stages of initiation, you can't really differentiate a nodule from a lateral root at the very earliest stages. So we wanted to ask, really what underpins this formation of a nodule, and exactly what is a nodule, and what allows a legume to activate a nodule that can accommodate nitrogen-fixing bacteria. So we took a comparative transcriptomics approach, comparing lateral roots to nodules using high-resolution spatial transcriptomics. And we across multiple time courses for the early initiation of lateral roots and early initiation of a nodule. And one of the first things that we observed was that, in fact, there's a massive overlap in the transcriptional profile between a lateral root and a nodule. 80% of the genes induced in a lateral root are induced in a nodule. So really, a nodule is a modified form of root development that's been initiated but then modified to allow accommodation of nitrogen-fixing bacteria. And consistent with that, we see that the key regulators that induce lateral roots, these are the LBD proteins that have been shown in many species to be the key genetic regulators that activate a lateral root. They are also induced very early in a nodule, and they're also essential for the very earliest stages of nodule initiation. So we know that the symbiosis signaling pathway activates cytokinin in the cortex, activates the transcription of this major regulator NIN, and then this NIN transcription factor turns on the program of making a lateral root. So this is apparently one key innovation in a legume, is that symbiosis signaling can turn on a lateral root organogenesis program, and that does not happen in cereals. But clearly, there's a big difference between a lateral root and a nodule. It's not simply that symbiosis signaling is turning on lateral root development. That root development has to be modified to accommodate nitrogen-fixing bacteria. And we wanted to know what were the key innovators that gave that novel form of development that allows association with nitrogen-fixing bacteria. And in our transcriptional profiles, one of the key regulators that stood out that was very surprising was this transcription factors LSH, which stands for light-sensitive short hypercotyl. And the reason it was so surprising is because LSH has been demonstrated in many plant species, rice, tomato, and Arabidopsis, to be expressed in the shoot, to be associated with the formation of inflorescences, and responsible for the complexity of the inflorescence that forms. It never been observed to be induced in a root context. And here we saw them coming on very early and very specifically during the formation of a nodule. Now, in legumes, when you mutate the LSH transcription factors, you have a massive impact on nodulation. The blue color indicates the bacteria inside the nodules and demonstrates that those bacteria are expressing the enzyme nitrogenase responsible for nitrogen fixation. And you can see in the LSH1, LSH2 double mutant, it completely abolishes the ability for those nodules to accommodate nitrogen-fixing bacteria and to transfer into this nitrogen-fixing state. So we believe that what's happening in legumes is that the symbiosis signaling pathway activates the expression of this master regulator, NIN, and that controls multiple developmental pathways, including a final step that gives you nodule maturation. And these three parallel pathways, activation of a lateral root program, activation of these LSH transcription factors, and a final step in nodule maturation, these three stages essentially transfers, first allows the activation of organogenesis in the root, and then transfers that organogenesis into a mode of organogenesis that can accommodate nitrogen-fixing bacteria. Now, all of this is the genetics that underpins nitrogen fixation in legumes, but none of it is novel to legumes. There's not a single gene on that program that has not been well characterized in other plant species and known to function in development in other plant species. And in fact, the transcriptional networks that we see associated with nodulation are consistent with how we understand these transcription factors functioning in other contexts of development. So what we're seeing in nodulation, as I said early on, is a pick and mix of development. You've taken our buscular mycorrhizal signaling pathway, you've hooked it up to lateral root development, you've hooked it up to a novel form of shoot development, you've hooked it up to a maturation program. It's not that you've actually evolved any new functionality. You've re-networked pre-existing developmental contexts into a novel mode of development that has allowed an innovation, and that innovation is a switch from engaging with our buscular mycorrhizal fungi to now engaging with nitrogen-fixing bacteria and activating the developmental context. Now, when you think about transferring an association from a legume to a cereal, obviously, if you have to build a signal transduction pathway and a whole developmental process, it's going to be well past my lifetime to do that. A re-networking of pre-existing developmental processes is a much simpler approach than starting from scratch. And the fact that everything that we're seeing in a legume is already present in our cereal crops, and that it's actually a novel modality of their activation that seems to be the key innovation, suggests, at least to me, makes me very hopeful that it's very feasible to transfer this association. Now, my biggest dream is that each of those transcription factors there controls the same transcriptional network in a cereal as it does in a legume. My greatest fear, and the thing that keeps me up at night, is that LSH in a legume controls 1,000 different genes than LSH does in a cereal. And I have to put 1,000 cis elements in upstream of 1,000 different genes in cereals. So we will find out. But it's very exciting that I can tell you today that we are able to engineer nodule-like structures in cereals. This is barley. These are inducible nodule-like structures induced by rhizobia. And developmentally, at least macroscopically, they look like nodules. At least developmentally, they look like nodules. Transcriptionally, we see a response that's not inconsistent with lateral roots because a nodule is a modified form of lateral roots. The one difficulty that we have, the biggest challenge, the red here on the right-hand side, is the rhizobial bacteria that we've used to induce those structures. And you can see that rhizobia is not getting inside those structures at all. It's staying on the outside of these structures, and it's not getting that intracellular invasion that's so critical. So while we are at the stage now in the project where we can control development in cereals to activate structures in an inducible way that are not dissimilar to nodules, we don't have a functional nitrogen-fixing nodule until we've got bacteria inside those structures. And so this final stage, yeah, I'm finished. This final stage, the stage that we're currently at, which is a lot of discovery science and experimental engineering, is really to understand exactly how bacteria get into those structures in legumes and to engineer this component part into cereals. So with that, I'd just like to finish. So our vision for the ENSA project is cereals that are able to engage with a much broader array and much more effectively with microorganisms, providing far more equitable and sustainable ways to fertilize our fields. And we work very closely with the photosynthesis projects such as RIPE, because there's a close interface between how much carbon you can take, how much nitrogen you can take, how much phosphorus you can take, and the costs as well of these microbial nutrient surfaces. They don't come to the plant for free. They cost the plant in the form of carbon. So we are a large program and multiple partners involved in the program, now funded by Bill and Melinda Gates Agricultural Innovations. And for the talk today, I just want to highlight two particularly scientists, Cash Schissel, who's a postdoc has finished now, has been a postdoc in my group, did a lot of the work on the development, and Frank de Tengu, who did the work on that engineering of nodulation in cereals. Thank you very much.
Thank you. Thank you very much, Professor Oldroyd. Time is fast spent. I think we have about 4 minutes to take a break. I will take a couple of questions, and then that'll be it. So I have 2 hands up. Please, can you help us with the microphone?
Thank you very much for a very interesting talk. I have to change my lecture now for the students because it's the first time I've heard that the mycorrhizae are not symbiotic, they are parasitic. So how do we do that? Can you explain so that I can convince them? It's difficult because we always taught them that it is symbiotic. That's the first part of my question. And the second one, when you talked about this maize line sensitive and resistant to mycorrhizae, you know that there are plenty of mycorrhizae, Glomus sp. Do you think of gene-for-gene interaction somewhere?
So I'll take the second question first. So those maize mutants, they're in the symbiosis signaling pathway, and they abolish all engagement with all mycorrhizal fungi. It doesn't matter which species of mycorrhizal fungi you use. We've seen no colonization by any mycorrhizal fungus at all. So there's certainly a lot of gene-for-gene interaction in the nitrogen-fixing symbiosis, where you have very specifically decorated lipoketoligosaccharides and receptors that can specifically recognize them, but we don't see evidence for that in the arbuscular mycorrhizal symbiosis. And the first slide, I think it's an underappreciated part of mycorrhizal. Everybody thinks it's a beneficial plant at all times. It's actually quite a conditionally beneficial. Under certain circumstances, it's beneficial, and under other circumstances, we should consider it parasitic. And there's some plant species where we've never seen benefit to mycorrhizae. But in rice, maize, and wheat, we've seen exactly what I just showed you. There are elite varieties we're currently planting in the field that are actually suppressed in their growth by mycorrhizae. It's probably that they're not delivering, they're taking a lot of carbon and not delivering enough nutrients in return. And in that case, when we haven't looked in detail at this, there might be a lot of variation for the actual fungus. So some species of mycorrhizae are more beneficial than others. And if you put them in one field context, you may see more benefit than others. So I think it's probably in part the plant and the fungus mix that drives the extent to which there really is benefit that can be accrued by the presence of mycorrhizae.
Okay. I think we'll take one more question from the gentleman up there.
Cannot hear.
Thank you, Dr. Oldroyd, for the wonderful presentation. My question is about breeding plants for improved mycorrhizal association. Like the professor, the doctor has rightly said, mycorrhizal association, it's actually on a mutualism parasitic continuum. Like, I mean, in certain contexts, you expect the plant derive more benefit, and in other contexts, the plant loses more than it gives to the, more than it gains from the symbiont. So how can we tailor our breeding? Of course, I'm not really sure. I really don't know any study that is currently considering breeding plant for improved mycorrhizal association. But in that case, if we want to take on that project, so how do we tailor our breeding program to make sure that the crops are bred to select more mutualistic mycorrhizal association in the field? That's
Very good question. Yeah. So part of the ENSA project is to actually look at those lines that are getting maximal benefit from mycorrhizal fungi and define the genes that control that benefit to provide genetic markers that breeders can use in breeding to improve the associations with beneficial mycorrhizal fungi. So I'm not aware of any breeders who are looking at mycorrhizal fungi. So we are in the process of trying to get that genetics of mycorrhizae established so that you can actually, it's very, very difficult to quantify. It's not an assay you can do in the field. You can do it with those mutants and you can see the benefit you get in the field, but it's not an assay you could very easily walk the field and say how much fungus or how much benefit the plant is getting from the fungus. So I think it's a trait you'd have to use marker-assisted breeding to bring into play. And I think hopefully, I'm hoping a combination of hypercolonization and the genetics of benefit, those stacked together will really drive up the benefit that we accrue from mycorrhizae and set you so that you're maximizing the beneficial association you have and limiting the parasitic association.
Thank you very much. Please give it up for Professor Oldroyd. Thank you. So we'll take our first break. But before that, I have a couple of announcements. The parallel session will take place at 10:45. The rooms are upstairs. You just have to go upstairs. You have 2 rooms on each side. You just follow the direction, and it will take you to whichever session you are interested in. Please, pardon? There's also one that is here. So, so there's also one that is here. I will hand over to Dr. Rufaro Madakatsi to give her announcement.
Good morning, everyone. Hey, good morning, everyone. Yeah, I wanted to see if we are still alive. As he said, my name is Rufaro Madakadze. I am the Electoral Commission of the APBA, and we are going to have... please clap for me. It's a big job. Thank you. We are going to have elections tomorrow, so I wanted us to just be clear what we are going to do. The role of the EXCO for APBA is critical. We need people who are serious, people who can link the association with other global partners, people who can recruit people, so that we start growing. We only started in 2019, and we have been on a growth trajectory. So the first EXCO has done a very good job. So please give it up for them. I know Eric has had sleepless nights. Your sleepless nights are going to end tomorrow. We are going to give it to someone else, so thank you so much. So we want to have nominations. So please send your nominations to, what is the email address again? We need an email address to send the nominations to. You can nominate yourself. Okay, I need to remove my glasses. It talks about age. The email is apbexconnom@wasi.ug.edu.ghana. Please, someone, can you put it on the thing so that we can, so we will email it to everyone so that you can nominate. The second thing is for the current EXCO members, you can only continue if you are vying for a different position. So we want to make sure that we have new blood. So please, this has to be a clean campaign. I'm from Zimbabwe. We normally we can't even contest elections, but these ones, these ones we are going to contest. We are going to run a fair and clean campaign, and it will be good. So please nominate people. Please nominate yourself. And if you are campaigning, let's keep it clean. Thank you.
Please, one more thing. So there is a desk at the registration area for members to pay their membership dues. Please note that you cannot vote if you haven't paid all your membership dues. Then there is also the poster session ongoing across the street. You can take your whilst taking your break, you can just cross over to the other side and then engage the posters. Thank you very much. There is a request by Dr. Rufaro Madakadzi. All participants who are sponsored by AGRA, please meet in front over here. All participants who are sponsored by AGRA, meet in front here. She also wants to meet all the plant breeders from Zimbabwe. All plant breeders from Zimbabwe, kindly meet up here, down here for a quick information. Oh, the wacky one here.
All right, so we can start. Hello, good morning. Good morning. Please, let's take our seats. Hello, good morning. We want to start the session right now. Please take your seats or move forward. I'm sure all the speakers are here. To go this way. Hello? If you're on this side, please move this way. I think you will see the slides better when you are on this side, because normally this thing blocks some of the slides. So if you're a bit on this side, I think you'll see better. We encourage everyone to come on this side. We'll start speaking now in the interest of time. So I'm the chair of this session. My name is John Derera. I'm the Senior Director for Plant Breeding and Pre-Breeding in the Genetic Innovation Science Group. So the main objective for this session is a communication session where we are going to be talking to all our partners and all of you here about what we do in genetic innovation and how we collaborate, and also how we can strengthen our partnerships and develop new ones. We'll also be informing all of you about how genetic innovation works and its current initiatives, and try to build a shared understanding of how we can work together and how we keep learning and improving at the same time. So in terms of speakers, we have 5 speakers representing various departments and initiatives who will be speaking in this session. Amongst them is our Managing Director, Sonia. She is here. She'll introduce Genetic Innovation Science Group and also give an overview of the breeding resources initiatives. Then Dorcas will talk about accelerated breeding. Then Brendan will talk about market intelligence, and Chris will talk about the seed systems, seed eco initiatives. And Professor Michael Aberton, who is here with us here, will talk about genetic resources. That's the gene banks and how they link together. So we have 2 speakers, followed by a question and answer session as we go. So without wasting much time, I will invite Sonia to come to the stage. Sonia, please.
Thank you very much, John. And I think I need no further welcome. I was speaking yesterday, and it's been a real pleasure to speak to everybody also over our tea breaks and lunch breaks. So I'm going to give a short introduction now to how we are collectively organizing our work at CGIAR on what we call genetic innovation, and this includes gene banks, plant breeding and pre-breeding, and the work on seed systems. So just first of all, to talk about the way in which since 2019 we've embarked on a new research and innovation strategy, and the attempt is to become far more integrated across our CGIAR centers, across crops, and across regions. And in particular, this is enabling us to be much more responsive to regional and national priorities and to work in more effective ways with partners. So this is the new strategy that I mentioned, and it has very specific targets that map to the Sustainable Development Goals. And so we're expanding the CGIAR ambition from speaking predominantly to food security to a wide range of ambitious impacts across gender equality, people's nutritional status, jobs and livelihoods, environmental outcomes like water use efficiency and biodiversity, and also, of course, climate change resilience. And so I've just mentioned the kind of 5 big impact areas, and then we work in 3 science groups to deliver those. So just over 2,000 members of CGIAR staff map to genetic innovation. That's our group, and many of us are here today. By speaking to us, you'll know that we work in many different centers and in many different locations around the world, with a predominance in Africa. And that's the group I'm representing today. And you will see that we have a very matrixed way of managing ourselves. We work in lots of different ways together. We are focusing on these strong country and regional partnerships. We actually work in 6 regions across the world, and 3 of those map to Africa. So there's Sewana, which includes the country we're in today, looking at Central and West Asia and North Africa. Then we have West and Central Africa, as well as the many colleagues that we have here also from East and Southern Africa. And we are trying to match our partnerships to specific challenges. I talked quite a bit about this yesterday, where we're looking at the problem solving, we're looking at what capacities different partners collectively bring to that, who's got the comparative advantage on different pieces of the puzzle, and then how do we fund that and deliver it collectively. Now I'll get into the kind of detail here of how we've organized ourselves. This little flower diagram shows how we are organized in genetic innovation. And just to emphasize again, this brings together personnel from all of CGIR's centers. So people in the audience or people you'll be meeting from CGIR will be working both for CIMMYT and within this arrangement, or with IRI or IITA, ICADA, and so on. So at the very center are our gene banks. We will hear today from Michael Aberton on those, but just to say, we also have in the room our global senior director on gene banks, Charlotte Lusty, who won't be speaking, but will be speaking with many of you during the day and during the breaks. So thank you, Charlotte, and thanks for nominating Michael to speak on your behalf. Then we have our work around market intelligence. We'll be hearing today from Brendan Rice on that. And Brendan is representing our global leads in that area, Matty Mont, who sits at IRI, and Vivian Polar, who sits with SIP in Peru. So that's a very new area of work, of really making sure that we're more demand-driven. Next, we have our actual breeding work. So at the center of this is accelerated breeding. And you heard yesterday from our lead on that work, Michael Quinn, and Michael has nominated Dorcas to speak on that area of work today, so we'll be hearing from her. So that's the really bulk of our work on taking forward crop improvement, and particularly trying to improve how we do it, get faster and better. Supporting that, we have breeding resources, which I'll introduce to you. Our global senior director in that area is Sharifa Syed Alwi. She's based in Asia, but leading globally on the infrastructure and data systems that not only us, but NARS need to make the breeding networks work better. We also have another breeding-related initiative that we keep a bit separate, and that's just for funding reasons. So we do work on genome editing. It's obviously an integral part of breeding pipelines, but we keep it slightly to one side because of European sensitivities around funding. We have secured a set of funding around the world for that, and will be taking forward not only a few key use cases to demonstrate how genome editing can make a difference, but also bringing together, convening the NARS around the world who are very interested in changing the policy dialogue on this and enabling all of us around the world to make progress, to get past some of the regulatory barriers we're encountering at the moment. Finally, and just to say, our lead on all of the breeding and pre-breeding work is this modest man on my right who's chairing us today, Professor John Durera. Finally, we have our Seed Equal work. This is around seed systems, and we call it Seed Equal for a reason. We're trying to achieve access, equality of access and equality of use in those seed systems. So, of course, we're looking at markets, we're encouraging formalization of seed systems where possible, but at the same time we recognize that many of the current seed systems around the world, and some of them being the most effective seed systems, are in fact informal. They're farmer to farmer, they don't involve formal markets. Our senior director on that area of work is also with us today. I hope some of you have spoken with him. His name's Ian Barker, and he has asked the lead of this specific initiative, Seed Equal, Dr. Chris Ajiwo, to speak on this area of work today. Chris leads our work on getting the seeds into the final systems and to farmers. Right, I hope that's the sort of big picture, and that flower shows our kind of global organization, and you'll be hearing from each of these teams. I won't dwell on this slide, but for those of you who are more pipeline-oriented in your thinking, just to say that all of the work that I've described, we have also mapped to the kind of pathway of innovation from the product design and market intelligence side through the product development, all the way from trait discovery through to the crossings, the screenings, testing at different stages, right through to the on-farm testing, and then finally the product delivery. That's the Seed Equal end of the work, product registration, and then going through the launch and so on. And our work supports some of the regulatory environment around that. So we map to this pathway, and we're trying to take not only germplasm through the pathway, but also capacity, thinking very carefully about how we build capacity throughout Africa at each of these stages. We have globally about 900 partners. That may make us sound unstrategic, but that's really because of the number of countries we work in. So our primary partners, our primary peers, are around 200 national agricultural research and extension services. Many of the others are either private sector, and then, of course, some of the funders, some of whom have a lot of technical knowledge, and NGOs that are active in particular situations. I'll move on from that, just to say, to plug again that on Thursday, we'll be meeting with NAS partners to review the relationship that we codified in an aid memoir last year. So we really are trying to get this integration across the CGIAR centers, market intelligence work, how are you identifying market segments, prioritizing them, how are you defining the target product profile that determines what you breed to, sharing methods on phenotyping, for example, which we heard a lot about from Michelle this morning, but also sharing outsourcing, sharing procurement when we, for example, go out to private sector to source genotyping services. Also shared data management, big data systems. And it's been successful, right? I mean, Michael was talking about it Michael was talking about it yesterday, the extent to which we've been able to reduce the mean breeding cycles really quite dramatically in recent years. Our gene banks also are real leaders in setting standards, meeting them, and building capacity amongst national gene banks to do the same. Okay, and this does lead to impact, right? So last year we had partners registering, I think we've used the word release here, but registering over 300 crop varieties across 40 countries. We don't do that registration work. We're obviously more upstream from that, but this is involving CGIAR germplasm. And I think critically, I spoke earlier to how we're widening our impact focus. So I think importantly, 3/4 of those varieties did have climate resilient traits, and 35% of them showed traits that are very deliberately bred in to speak to the malnutrition issues amongst women, adolescent girls, and children. So I'm going to change tack now and dive into one of these areas. Sharifa Syed Alwi, who leads this work, is based in Asia, and she's at the International Rice Congress and wasn't able to come over, so she says hi and sends regards to everyone. So I'm just going to take you through the kind of work she does with Enquang and many others around the world. So Sharifa herself comes from the private sector, and she's got a lot of ideas on how to do things better, faster, and cheaper. And the idea of doing things better, faster, and cheaper in our world is not about maximizing our profit. It's about getting a wider set of meaningful choices to farmers a lot faster. Climate change means the challenges are coming thick and fast, and so we're trying to keep up with that pace. So this is what our hypothesis is: the better we do internally, the better our external offer. So these are the set of work packages. I won't go through these in a lot of detail. You can see the text here, but it cuts across service provision, operating procedures, data management, which for those of you who do it know is a big monster in budgetary terms, but is really worth doing. And then, of course, all of the infrastructural investments. And when we're looking at these, we're not just looking at this for CGIR, right? I spoke yesterday about working in breeding networks, so we're looking to raise these capacities more widely than just in our own centres. So here's an example of that. CTEH, you'll see that acronym there, stands for Crops to End Hunger. In this case, it's a specific investment by Germany. They put in $15 million last year, and they said, put together a prioritized set of investments that are needed to raise the critical capacity of breeding across Africa within these breeding networks. So this is what we did, and put out a set of competitive bids, and you'll see here these important investments in the capacities at IITA, CALRO, NARO, Africa Rice, and so on. So these really are around fulfilling basic infrastructural needs to take the work forward. Someone may be able to share some good examples later. Going forward, future outlook. The first future outlook is actually coming tomorrow. So on tomorrow, a service portal will be launched, a one-stop shop to access all of the shared services available, developing an analytics pipeline, and then to really expand the kind of thinking on standard operating procedures across a wider set of processes. And what goes hand in hand with that is some training. We are trying to be smarter about training, do less supply-led training, and more going to people and seeing what the needs really are. So the offer list is long. You can see the ... continuous improvement ... but that really provides a menu that we can then map to users' needs. And here are some people with some examples. So I'm making a guess now that one of the new equipments here has actually been some tractors for some of the field work. And there have been examples elsewhere, the investments in Ghana, for example. So I think I think I've reached the end. So thank you from me, and I'm going to pass on to a colleague for the next segment. Yeah. Do you want to take questions first?
No, not now. We'll take questions after the next speaker.
Okay.
Yeah. Thank you so much. May I ask Dorcas to come through? As Dorcas is coming, I can just say Dorcas is our leader on quantitative genetics. She leads that team across the CJ teams. And she's also the work package leader for the Accelerate breeding. And she works under Michael. Dorcas, here you are.
Thank you. Mabibi na mabwana, mesdames and messieurs, ladies and gentlemen, I'm going to talk about accelerating breeding at CGIAR NARE's programs. And, okay. Okay, doesn't go that fast, and it goes too fast. Okay, this is according to the vision and the support that's being provided by the Accelerated Breeding Initiative, the one CGR. I'm Dorcas Gemenet. I lead one of the work packages in this initiative, and the initiative lead is Michael Quinn. He spoke to us yesterday. You can catch him during lunch also for more questions. And the initiative is focused on helping the CG and NRS breeding programs to deliver higher rates of genetic gains in terms of farmer preferred or end user preferred varieties in a step change way, because it's totally impossible to change everything at one go. And therefore, this initiative is focused on modernizing the CGA and NRS breeding programs. As I said, of course, the lead is Michael Quinn. And we are mainly focused on enabling better outcomes, for example, in the CGA NRS networks, to have joint priority setting for what we breed for. And this, as you heard yesterday from Michael's talk, would enable strategic use of resources and be able to create more impact with the resources that we have, whether they're human or financial, and also to mainstream best practices within these programs across the CGA and the NRS programs. And in this way, then the CGA and the NRS are more collaborative, and they are able to share capacity to deliver more nutritious, climate resilient, and gender responsive market demanded varieties. This will then be able to enable our farmers to be able to replace the old and outdated varieties, so that the new varieties which have been bred to target the current environment can help the farmers, I think, to mitigate the challenges of the climate change. And with this, we are hoping to sustainably be able to meet 50 to 60% greater food demand. So, I didn't delete one slide. Sorry. That was an edited one. So to be able to achieve this, of course, we need key partnerships in place, as Sonia just mentioned. And some of this detail you saw in the last presentation as well, that we're working with a little over 1,000 crop-specific collaborations with about 600 institutions in 136 countries from 36 crop by region networks. And these institutions are about 38% NAS, 23% universities, as well as private sector. You saw Bayer being represented here yesterday. And we are focused on countries, our main emphasis being in the low income and low to mid income countries. But there are interests also from the upper middle and high income countries that also are participating in this and benefiting from this CGR collaboration. So to deliver this, we have commitments, high-level commitments via work packages. And today I'm representing my colleagues, some who are not here, but some of them are here. And we arranged in 5 work packages. The first one is called Refocus, and is supposed to help us meet the farmers' needs based on the information that comes from market intelligence. And this is the work package that links ABI to market intelligence, so that we are able to align breeding targets with the breeding efforts. The work package 2 is aimed at standardizing things across the breeding networks to organize the activities so that we can have clear stage gates on where clear decisions can be made, who is having a higher role here, who is having a supporting role, what are the KPIs and things like that. It's called Reorganize, and it's led by My colleague Sarah Han, who was not able to join, Peter Coldrake for Work Package 1 was also not able to join us this time. The Work Package 3 is Transform, which is aimed at building inclusive and impactful partnerships, which is also related to the meeting we'll be having on Thursday, and that's led by my colleague Bish Das. He's in the audience, and you can catch him over lunch if you have additional question on Transform. Work Package 4 is called Discover, and it's supposed to help us drive, or it is helping us to drive and improve trade discovery and deployment, especially also aligning these high-value trades with the breeding targets and not just going in without prioritization on where we focus and invest our resources. Work Package 5 is called Accelerate. It's aimed at optimizing breeding pipelines to accelerate genetic gain. That's the work package that I lead. And if you have any questions, please catch up with me. So as we said, this is a breeding work package, so we are all in it. And these are some of the things that we have achieved so far. You saw some of this in Sonia's presentation, that in 2022 alone, about more than 300 varieties were released through the CGR and RS networks. And we also have now the first ever portfolio of breeding pipelines. And as we said, from Refocus, we have a breeding portal where you can have this information open access, therefore promoting transparency on what we are investing in. And this enables the managers and the breeders to make better decisions in terms of resource allocation and the germplasm they are working on to ensure maximum impact. We do have now a common framework across crops, like I mentioned, through Reorganize for clear stage gates. Where is market intelligence taking the leading role? Where is breeding taking the leading role? Where is seed systems taking the leading role? Of course, each of those components should be linked to the next and know what is happening in the prior part, but the extent of roles differs, and these clear stage gates help us to improve the efficiency and also to come up with the key performance indicators at each of those stages. And then from Transform, of course, we have had of the aid memoir that was signed, I think, last year. And that's related to the meeting we're having on Thursday. So if you're invited to that meeting, of course, we'll be happy to have you there. But of course, this is focused on improving our partnerships as we move along, because this is important to deliver on this ambition. We do have now the first ever view of the trait discovery and deployment work that is going on within the CGI and NAS networks, which information we previously didn't have, which traits are being prioritized, how do they relate to the traits that are needed in farmers' fields and things like that. And then, of course, we have been supporting optimization of breeding schemes, also providing quantitative genetic support, analytical support, and such through Accelerate. There have been large investments from the GIZ in terms of crops to end hunger. Sonia just mentioned that. Even though that's an investment that has come from breeding resources, its main focus is, of course, to improve the breeding operations that can improve the efficiency of the breeding programs, the accuracy, and things like that. And then, of course, we have developed work plans for modernization that we are working through. We have the ABI work plan that most breeding teams are working in. So just an example of highlights of our work in Africa. Of course, we mentioned we have several crop by region networks. So we have 55 crop by region networks. 26 of these are actually based in sub-Saharan Africa, and this indicates the focus and the effort that we're putting into attending to some of those things that were raised by our colleague Mpa yesterday from FAO. And within these partnerships, we're working with about 100 national programs and small and medium enterprises in about 30 countries. And working with these partners, we're also trying to understand what are the preferences by doing consultation with all stakeholders. And we have had about 20 consultations this year through Transform. And of course, we have mentioned the infrastructure that has spent about 15 million investment in several centers within sub-Saharan Africa. And this supposed, of course, to improve trial quality, data quality, and things like that, that we are interested in improving our efficiency. We have also worked with the partners to try and expand the on-farm verification of varieties so that before they go out, it's already known if they have what it takes according to the blueprint that the farmers and the end users have given. For example, in Tanzania, TARI moved from 20 farmers to 1,000 farmers, which is a great expansion to be able to capture more farmer preferences on the varieties that are being developed. So moving forward, of course, we do plan to continue to work together on accelerating the rate of genetic gain, and of course, prioritizing what we invest in and how we design our products and our breeding targets, and to generate regionally available germplasm, which of course is our main focus. We plan to continue to do program and infrastructure assessments so that we can be able to know where more support is needed in terms of capacity and other kinds of support for modernization, to provide technical support in quantitative genetics, costing, among others, and to continue connecting, putting strategy together and sharing. And that is, of course, for example, our meeting that we'll be having this week, and to facilitate, for example, the CTH grants to make sure that they actually give impact on the breeding programs. So with that, I want to thank you, and we'll be happy to take questions.
Thank you so much, Dorcas. Maybe you can remain here for the questions. Our next speaker will be coming is Brendan, speaking on market intelligence. So for now, I can take 2 questions for the 2 speakers. Yes, how do we get the mic to you a bit far? It's up there.
Yeah, I'm Rashid Siraj from UM6P. Very nice presentation, thank you very much. Both last presentation and the first one also from Sonia. And also impressive numbers, 300 varieties released 2022. My question was more, I don't know, you might have the data, but it's more about the intake of those varieties. If we have some sort of M&E, monitoring evaluation, what kind of traits, what kind of impact? Because obviously the number of varieties released is one indicator, but of course if we want to look at plant breeding, especially in Africa, we're going to see what kind of impact pathways we're shooting for. So if you have any kind of evidence on that, that would be very much welcome. Thank you.
Yeah, thank you very much for that question. I think that's the bigger question from the presentations we have had since yesterday. Michael, do you want to take that? And I think Chris, who is coming next, will also talk about the seed system part of that.
Sure, thanks, Dorcas. Fully agree with you that that just number of varieties is not a strong metric, particularly on its own, and we need to understand, well, what is the impact and how good are they? So first of all, I mean, we do put it there to show that the system is working. I mean, there are varieties getting out there and getting to farmers, which is I mean, we need to ensure that the system's working to know that we can have impact, but also we're putting in systems to better understand performance on farm and working together with market intelligence and Seed Equal to understand the impact as well. So it's one part of the picture. It's not intended to be the whole story, and we are working at better understanding what happens thereafter, being the impact.
And I can just add here, Rashid, thanks for a great question, and you're jumping ahead to our other groups, right? So what we think is the best way to proceed is to have monitoring and evaluation at the level of all of our work. It even includes the monitoring and evaluation of how our gene banks are performing. And of course, some of their germplasm goes into our breeding pipelines and others goes out to the rest of the world. So we have a program that are doing that. They're doing a series of in-depth, gold standard impact studies. And in addition to that, we have a number of additional tools just to track things such as adoption rates. So there's a very smart app, for example, that we're working with. It's called VAR Scout. And what it does is it enables field workers to actually just register online. It's connected to a GIS system of where different varieties are being recorded. It's enabling us to really improve our ability to have real-time, real-place tracking of where adoption's occurring. So that's one tool we're really excited about, and we're working with NAS on it in a couple of countries. Ian can shake his head if I'm wrong, but I think it's Mozambique and Ethiopia. Kenya, Peru, and Ethiopia. Thanks, Ian. Okay. Yeah, so great challenge. And as you know from historical work, we do have very good examples of impact from CGI crops, and particularly on nutritional outcomes as well as on incomes. Thank you.
Thank you so much. I can only take one question. We'll have another opportunity later.
Yeah, there are too many answers, right?
Yeah, just one. There are so many answers to this
Hello.
one.
Wallace Cowling. So it's a very simple question. You mentioned resources for breeding and partners and trying to find this balance between funding breeders in the CG system and partners. How do you provide security for a breeding program? Security for breeding needs to go ahead 10 to 20 years in order to develop a good result. How will the CG system provide security for its core breeding programs? Quite a simple question.
Very simple to ask and perhaps not so simple to answer. I wish you were asking that to our funding community, Wallace. Yeah, so, I mean, we're doing our best. CGIR, as you know, has a trust fund. We ask funders to raise their level and put in money into a trust fund at a collective level, because that then enables us to do two things. One is, within a year, prioritize it as effectively as possible. And then the other part of it, of course, is to try and provide, if not stability, at least some year-on-year predictability. We're not nearly as good at that yet as we'd like to be. But yeah, we're working towards it.
Thank you so much, Sonia and Dorcas. Brendan, please come through and give a presentation on market intelligence. So what we'll do the same thing. We'll have two speakers. After Brendan, Chris will be coming right away, and then we take a few questions in between. And then finally, we move on to the gene banks. That's the modus operandi. Please write down your questions. We'll also, at the end, if we save some time, we can still take general questions at the end. Brendan.
Thank you. Hi, my name is Brendan Rice. I'm a research analyst with IFPRI, and I'm presenting today on the Market Intelligence Initiative, stepping in for Julius Okello, who couldn't be here today. And I would like to start with the next slide, touching on the vision of the Market Intelligence Initiative. If we could just move to the, I'm so sorry. This is self-driven. Okay, so the vision of the Market Intelligence Initiative is to maximize the investment returns in breeding across the 5 impact areas of the CGIAR. And this is about developing market information that can inform the work that breeding does and to improve this demand orientation of breeding activities. Because of the sort of nature of how market intelligence is conceptualized, it brings people of different disciplines together and also different institutional affiliations. So it's conceptualized as a way of developing co-ownership in breeding efforts. In market intelligence, also, this initiative is also important for feeding into the priority setting of the genetic innovation work generally that was presented at the front. So what is market intelligence? Market intelligence involves the practice of gathering and analyzing data on market demand to optimize contributions to the 5 CGIAR impact areas, which you can see on this slide here: nutrition, health and food security, poverty reduction, gender equality, climate adaptation and mitigation, and environmental health and biodiversity. And also the contribution to these impact areas, we want this to be measurable. And an interesting aspect of the Market Intelligence Initiative is this work is actually built into the initiative itself in Work Package 5, where we're looking at what are our impacts towards these impact areas. And here's the structure of the Market Intelligence Initiative. Yeah, there's a lot going on, but I'm gonna walk you through it here. So Work Package 1 is about market intelligence, actually developing, compiling market intelligence, and having some best practices for doing so towards the goals of these impact areas that I've mentioned and the SDGs that you see on the screen as well. This is then fed into Work Package 2, which is the gender intentional target product profiling work. And this is about developing standard operating procedures for target product profiling and sort of looking into how market intelligence is adapted and integrated into the TPP development process. This then feeds into Work Package 4, which is how do we make decisions between target product profiles, between market segments? As you know, there are many market segments, each with, not each, but many with associated target product profiles. And it's important to understand, well, where do we put our resources in these towards having the greatest impact possible towards the 5 impact areas that I mentioned. And that's what Work Package 4 is all about. The sort of public front-facing aspect of this is the investor dashboard, where an investor can look at sort of which target product profile, which to move forward, which varieties will have the most impact in terms of where to put resources. Of course, it's important not to overlook Work Package 3 in the middle there on behavioral intelligence. This is looking into what are the determinants of adoption of varieties, what drives use among farmers, and then feeding that information into Work Package 4 on sort of deciding which to move forward, which target product profiles to move forward. And moving on to Work Package 5, this is what I was mentioning earlier about kind of this built-in impact assessment aspect of the Market Intelligence Initiative, which I think is interesting and is what I spend my time on. And this is about understanding, okay, what is the impact of market intelligent varieties with respect to not, just kind of normally developed varieties that perhaps don't take into account market intelligence? Are these varieties having an impact on varietal turnover? And of course, these impact areas that I've mentioned before. And then also, how do we scale this institutionally in terms of use of market intelligence in these processes of breeding? I've essentially covered each of these work packages already, so I'll just go very briefly through them. Work package 1, again, is about developing the market intelligence. Work package 2, integrating the market intelligence into target product profiles. Work package 3, behavioral experiments, what's driving use of varieties. Work package 4, I especially want to draw your attention to this work on the GLOMIP platform because it's out. You can search for it and take a look at it. This is about looking into impacts. There are a lot of impact indicators, and you can break it down by crop and location, and it's quite the tool, and I hope you take a look. Work package 5 is about experiments to understand the current incentive mechanisms that drive use of market intelligence, looking into sort of what's being done in terms of incorporating market intelligence into breeding programs, what information is there, how formal is it done, and so on. So we really want to get a baseline of this, and for this we really need your support, and I'll get to this in a moment. Right now, in fact, which is at the booth directly to the right when you walk in the building, there's information on 2 tools that we're doing now in Work Package 5, a survey, which is to look into how breeding teams collect market intelligence, sort of what are some incentive mechanisms within breeding teams, what drives breeders, and so on. And then also what types of information is missing, these sorts of things that we really need your support on in order to pilot this tool and kind of get at a version of the survey that can sort of function towards the ends that we need it to in terms of having better market research that that allows breeders to align their work with market demand. So this is really important. Please, at least I find it very important. At least please come by and grab a flyer at the entrance of the building, or scan the QR code directly. That would be a huge help. We also have some core innovations that I wanted to highlight. You see on the left here, standard for gender intentional TPP design in crop breeding. This is something that we're working on. The GLOMIP platform is what I've already mentioned, and I just want to go into a little bit more detail on it here. So as you can see here, there's a bit of information here, but the main takeaway is there are 173 sort of impact opportunity indicators. And you can look into these indicators across all the crops you see on the, well, you don't see the crops themselves, but just all of the crops that are available there. Countries, and there's a lot of visualizations that you can utilize to look into sort of impact possibilities in these efforts. And to go into a little bit further depth here, just to show the breadth of information in terms of these indicators, just to give nutrition, health, and food security impact as an example, there are 42 indicators. For gender equality and youth and inclusion, there's 43. And the same goes for poverty reduction for these 3 other indicators that you see on the screen now, and the cross-cutting indicators as well. So this is a tool that I hope you take a look at. And to go into just to show some of these visualizations, the columns you see there are these indicators, and the impact potential is broken down by crop. So there are a lot of partners research partners, scaling partners like the WFP, and also rigorous testing of these academic institutions you see on the right. So thank you very much, and looking forward to your questions.
Thank you so much, Brendan. And Chris is coming here. He's the leader for the Seed EPO initiative. And most of you know him in the seed systems. He has been in it for a long time. So he will give an overview of our Seed EPO activities. Chris?
Thank you. Thank you. I'll take you quickly through the Seed Equal initiative, whose objective is to support the delivery of seed of improved climate resilient market preferred nutritious varieties. Sort of a mouthful for that, but let me just put emphasis on one fact that I cannot, unfortunately this is not a pointer, but I would like you to see what I have indicated there as seed and what is indicated inside as improving and packaging genetics in it, and to mention in a single sentence that the emphasis here is to mention that seed is the vehicle that carries the improved genetics from breeders into the hands of the farmers. So for us to be able to create the impact that was being asked about earlier, unless you're able to deliver the seed into the hands of the farmers, then the impact will not be created. So with the emphasis on accelerating the varietal turnover that is coming from the accelerated breeding, so to say. And designed and arranged into work packages, just like the rest of the other initiatives. The first three work packages, more or less designed in sort of crop archetype, where the crop archetypes here around the cereals, the legumes, and roots, and tubers, and bananas. And the next four are handling specific issues. One is coordination for impact. This is where we have adoption and impact assessment in terms of design and implementation. Seed policy and enabling access, equitable access, particularly this is related to gender and related issues. And finally, emergency seed, which I will highlight a little later as strengthening resilience generally, because while initially designed as an emergency seed, it's not necessarily overall being handled in the strict sense as emergency seed. And over to highlight again back the first three, which were crop archetype based, I would like to mention that the intention is not to do a push, where I think the seed systems in many areas have been designed to move from breeder seed to basic to certified and then to grain. But otherwise for us, we would like to start with the grain side and say, liaising very closely the market intelligence to establish the level of demand, the current demand, the demand trends, the foresight, to be able to tell us in terms of product quantities and product quality that are being demanded for these specific crops and varieties, that then enable us to have informed decisions on what sorts of quantities are being or should be delivered, so that our supply, our delivery is more or less a response to demand signals rather than a push per se. And working closely with partners is one emphasis that I would also want to mention. This is where the work package number 4 is overall designed to take action. We've worked very closely with partners from the national as well as partners from the private sector. But I want to highlight a few partners around demand, innovation, scaling partners. Some of the partners that have of late been of interest to us and have interest also in us is International Seed Federation that has reached out to us to sign MOUs. We're working on similar MOUs with AGRA and similar MOUs with African Union that then handle across private sector as well as public sector in terms of ensuring that there is efficient delivery. And some of these are at final stages of signing contractual agreements or signing MOUs with these partners for efficient delivery of seed. And mentioning about policy solutions, again, there are several examples that I could cite, but let me just mention about plant variety protection laws that were not existing in Nigeria, that we worked very closely with New Markets Lab to sort of at the stage of finalizing plant variety or PVP laws. Accelerated private sector engagement example in Rwanda, market-led innovation seed distribution in Uganda as an example, and policy performance in Uganda's seed sector. We could have these cited from a publication. But more recently, that I would like to highlight, and that was highlighted in our 2022 key results story, was the advancing of the regulatory change for vegetatively propagated crops in Kenya. This has moved to a step where we are almost finalizing this into a law, into a revised policy that enables vegetatively propagated crops to be handled more or less through QDS or standard seed that currently is not allowed for any other crop in Kenya. And still closely related to policy is an issue of handover varieties for commercialization from breeding. And what has been successfully, I would say, promoted in the past is through an approach that involves consortia. And there are several consortia that I would cite: the Hybrid Rice Development Consortia, the International Maize Improvement Consortia that is with CIMIT. And of course, we've had several other consortia mentioned here. The Hybrid Parents Research Consortium for sorghum and for millet, again supported, popularized by ICRISAT. And recently, much more recently, AFSTA, African Seed Trade Association, has reached out to us and saying, we have a recently signed agreement with World Vegetable Center, and we like their consortium approach. We are studying that consortium approach and saying, how do we improve variety adoption, access, handover from research to private sector, particularly through this consortium approach. And closely related to the same, maybe before I handle this slide, just to mention the aspects of licensing that are also critical to us, and the approach to licensing has been, we've been doing a review across the various institutions, CG institutions that have been handling licensing. And the latest of the revised document around a common licensing agreement and approach just reached me yesterday. I'm reviewing it, and I'll be able to share feedback, but we are more or less at a stage where we are almost escalating it to a level where it could be approved as a CGI document for licensing. But at the moment, I just want to mention in terms of reaching the underserved customers, or the farmers, especially the youth and the women, or the work that is being handled by work package number 6, popularizing and inventing through what they're calling youth and women quality centers, currently being handled and pushed by Jumai. And without struggle on what that diagram explains, let me just mention that the focus here is to ensure that there is the right quality of inputs and the right quality of outputs. The right quality of input here, our emphasis being seed and linking up the youth and women in these quality centers to the sources of good quality seed, but also then downstream linkages to ensure that whatever they produce in terms of grain ends up in the hands of the right grain users, in this case, processors and off-takers. So it's an upstream and downstream linkage, particularly for women and youth actors. And on to mention on what I would say constitute our work package number 7 is an emphasis that started more on relief seed or emergency seed, that was designed with two broad objectives. One is around policy to support the same, and then the second is the technical support. And the technical support, where I want to put emphasis here, is on the aspect of the fact that if seed is delivered by institutions that are not necessarily involved in seed production and seed delivery, seed production or variety improvement, they would require a technical support in identifying the right agroecologies for the right varieties. But at the same time, as we work with them, and more recently working closely with the World Food Program, they have reached out to us and asked us that, yes, we appreciate the support, but we'd like a little more support in terms of handling value chains. And so we are working around handling value chains. That's what I was mentioning, that rather than put emphasis on emergency, we are also putting emphasis on relief and on value chains as an approach to ensure that these the right varieties are available with the right partners. Overall, I would say our work around quality seed is on robust policies that ensure that there is inclusive pathways to delivery and that the public and private sectors are involved, and that there is evidence-based metrics to evaluate our performance in terms of adoption and impact. And because John is up, I'd beg to stop here. Thank you.
Thank you so much, Chris and Brendan. We have time to take at least 2 questions before, as Michael prepares to come and lead us on highlighting what we are doing in the gene banks. 2 questions, please. Any questions? Yes. Scarlett.
Thank you very much. My name is Tony Oboa from Uganda. I'm a soybean breeder. I wanted to find out what has been the arrangement between CGs and private seed companies. Is there some kind of registration for the varieties? And are there monetary remunerations? Thank you.
Thank you. So second, should I just respond to that? Okay. I mentioned about the consortium approach where there has been a membership to the consortia, and members of this consortia have a privileged access to materials that are being developed before they are actually released. So with that, they are able to identify the right material for their purposes. If I take, for example, for hybrids, then they're able to identify lines that they'll use for their hybrid combinations, for their own I would say, variety development processes, commercialization, so to say. And that involves definitely a membership fee. And that is what I'm saying, what I said a moment ago, that International Seed Federation is saying we have sorts of these consortia with different institutions. Can we have a more umbrella-based consortia that all of us as institutions within the ISF would be involved? But also, AFSTA, African Seed Trade Association, is reaching out to us directly. So we are in a discussion that involves AFSTA, APSA, ISF in terms of engaging the private sector. But that does not mean that there is no bilateral licensing approaches that include an institution, a private institution, private sector institution reaching out to one of the CGIAR centers and saying, I would like to get a license for these particular varieties. Thank you.
Thank you so much. Okay, the last question. Michael, please come forward. Go ahead.
Yeah, thank you for the nice presentation, both of the presenters. My question is going back to, in the previous presentation, we talked about farmer-preferred varieties, where we stop. And here, Chris is more explained of market demanded. And then the market intelligence is there. Where do we stop? Are we going all the way to the value chain, so that it will connect to the farmers, connect to consumers, to the industry, so that the full value chain works? Otherwise, if we are talking about farmers, preferred varieties, we are not looking to the other half, which is a market pool. Where is the market intelligence in situations where there is no grading system, even in some of the countries? How we handle that part of the market intelligence. Thank you.
Thank you so much.
Okay, I'll try to go first on that. If I understand correctly, it's is market intelligence going back to the level of kind of downstream to the farmer, or to include also processors and other actors? Is that correct? And as I understand it and how the initiative operates, and I might lean on others to come in as well, but market intelligence brings together all of that information on, it's not just farmer preferences, but also processors operating in a specific area who might have certain, for example, dry matter content requirements and potato for chips processing and things like that as well. But also even forward-looking sort of foresight work on future demand through some models looking into sort of the effects of future climate shifts and so on. And then, so that's like market intelligence, and then that's provided into the process whereby teams putting together target product profiles sort of weigh those various aspects, and decisions are made in a sort of with co-ownership, as I mentioned before, sort of bringing together different disciplines and so on. So that's my first go at your question. So.
Okay.
Yeah, I could add briefly, perhaps Zodi, you remember Sonia's presentation that outlined the whole of the genetic innovation impact pathway or theory of change until the end, that market intelligence would come in, for me, I would say at two critical nodes. One is in the product concept, identifying the right market segments, and then developing product profiles that meet those specific market segments, so that the varieties are not developed without market in mind. And of course, market here is not just the farmers, but market is, I would say, post-farm value chain actors too, and that include consumers, that include processors and traders and others. But while that role of product design sort of feeds back in terms of feedback loop to breeders, the other node that market intelligence then comes in for us as seed systems is for them to be able to give us information, relevant information in terms of what sorts of demand are we looking at in terms of the customer profiles that we are addressing, the product profiles, the particular market placements for these varieties. And when you talk about market, it's not necessarily always the formal market, but quantification of markets could include as well informal markets. And I think that's where your concern is, that where it's not structured, how do you get that value? Thank you.
Thank you so much, Chris and Brendan. Thank you so much. We move on to the gene banks. And Michael is here to lead us on this discussion. Over to you, Michael.
Thank you, John. And thank you to the organizers, and apologies that you have got me, unfortunately, instead of Marie-Noël. I want to thank my co-authors, Charlotte, Mariana, Alice, and thanks also to Scarlett for getting things organized. So yes, let's get going. I think we all know that in addition to the climate change crisis that we hear about every day, we also have a loss of biodiversity crisis, which is just as serious, but we don't hear about it quite so much. So, of course, the Gene Banks initiative is really a part of that, and is perhaps a little bit wider part of that than we realize. We do have those 20 staple crops, but we also have many other species in our collection and a lot of diversity associated with that, including the wild relatives. And as it says there, the objective is really to really facilitate not only the conservation but the use of that germplasm, access to it, of course in accordance particularly with the international treaty. We think about access and benefit sharing, and a lot of policy work going alongside that. So we focus on that, conservation, really developing the tools for conservation for now and into the future, cryopreservation, germplasm health, I'll come on to a bit later, but that's an important part, obviously. We need to be able to move germplasm around, looking at ways to enhance seed longevity. So these are really research aspects in which we're optimizing the work that we do. Of course, the use of this germplasm and being able to make it more accessible, more useful to breeders through a range of approaches is very important, and that linkage to breeders inside the CG and outside. I'm not going to be talking too much about that because there's going to be a talk from Zach, the gene bank manager of ICADA, later on in this meeting, which really covers a lot of the aspects to do with use. And of course, the CG, you know, there's a limit to what we can do, obviously, in terms of conservation and use. So we have to do a lot also with national systems and other gene banks around the world to really try and develop this global approach to conservation, to distribution, and to use. So here's an outline of some of the achievements up to last year. We have this progress of the gene banks towards performance targets. A number of them are already there. We have a lot of work on capacity development and really tightening up. I think Sonia mentioned at the start that, you know, there's already been a lot of collective work over the last 10 years or more. You know, we had the CRP, we had the platform. The gene banks are really very well used to working together, driving up quality management, those aspects, SOPs. Again, the georeferencing, the genotyping, those are very crucial to our work in enhancing use. Zach will be saying more about those. Distributions from the CG gene banks is really a key factor in the movement of germplasm around the world, and likewise the work of the germplasm health units. So policy, as I mentioned, is an important component of that. That is situated within the initiative, and probably a lot of you will be aware that, you know, there's a lot of debated areas in there about DSI, about the future of the multilateral system. It's very important that the CG has a strong voice in there in representing ourselves. We're not obviously one of the parties, but we can play a very important role nonetheless. The gene banks, the conservation, the characterization, germplasm health. So those are all the components of the initiative. So the work of the GHUs is very, very large, as you can see here, and it's really, obviously, it goes without saying, it's essential to the movement of germplasm, so it underpins the work we do in terms of not only the gene banks, but the breeding and other work where any germplasm is basically moving around. So if we look at, we can also monitor, there's a lot of, we heard about monitoring earlier, certainly in terms of the gene bank, yes, there's a lot of monitoring of the work that we're doing in terms of what distributions is going along, in terms of what progress we're making with our collections, in terms of digitizing images, georeference data, I'll come on to that a little bit later. It's very important in terms of some of these allele mining approaches which we're now developing, subsetting, phenotyping, capacity breeding, and again the policy aspects. So building capacity, we've done a lot of this. It's one of the work packages within the initiative is really devoted to building capacity. This is a picture, the main pictures of actually IITA staff. So improving the composition, we're working with national gene banks, we're looking at their operations, we're looking at their collections, we're looking at how we can work together in terms of genotyping, in terms of safety duplication, and so on. So the way we're doing that is a very interactive one. We're looking at what those national gene banks have, we're looking at what they express as needs, and the sort of consultations that we have regionally, for example, within West and Central Africa. So an example from ICADA in terms of some of the use of genetic resources is this collaboration in terms of wheat really being very able to withstand heat and drought. So this is coming from a wild relative of wheat, which has obviously been conserved and characterized in the gene bank. In IITA, we have 6 of those staple crops that were mentioned, and we're working closely with the breeders on those, but we also have some what we're calling underutilized crops, for example, African yam bean, where we're really developing the work and the interest which will lead to these being able to play a bigger role in the future. And I think personally it's very good that at this meeting we're seeing that emphasis on the underutilized crops in terms of parallel sessions devoted to them throughout, and we are beginning to see also more interest in the funding, in the funders coming along and looking at these crops as we realize their importance in terms of climate resilience, in terms of nutrition. An example from Africa rice is again the use of phenotyping combined with genotyping, working with NARS, so the genotyping includes whole genome sequencing and so forth to develop subsets which are genotyped, to develop mini-cores, and to go forward to work on gene discovery and association mapping. So for the future, we're looking, as we said, we've already worked a lot together. We're looking to enhance that and to enhance our ability in one respect to give a very good service to those who are requesting. So we're talking about inventory management harmonization. We're talking about development of a portal for all requests. So we're really developing something which is going to be smarter, more user-friendly, more interactive, that better meets the needs of the people that we are distributing to, and is somewhat more proactive in trying to encourage the use of our germplasm. There's a lot also happening, as I mentioned, on the use, but that will be covered in Zach's talk later. So I think that's it. I thought there was a thank you slide, but the thank you is gone. But thank you anyway.
Thank you, Michael. I think you can sit here. Oh, wow. Okay, so Michael and all the speakers have saved about 5 to 10 minutes. So I'll be taking questions for Michael and any other speaker who has spoken here. They are all here. Yes, Michael first, definitely. We'll start with Michael and then any other questions. Yes, there's one here.
Okay, it's working. Thank you for the nice presentation. From the gene bank perspective, what is the plan to reduce duplication, redundancy in the gene bank, possession, and to identify traits of importance to the breeder and to make integration? Otherwise, having a genetic resource sitting in the gene bank and doing all this, very hard work does mean nothing. So what is the plan? What is the future? And what is the integration with the breeding program also?
Well, as I said, you'll hear a lot more in Zach's talk, but I think that I can say what you're saying is obviously correct. That's a major emphasis of our work. So particularly on the clonal crops, for example, in IITA, cassava, yam, banana, and plantain, yes. We have deployed genotyping, for example, to identify redundancies, to identify things which may not be true to type. And I think that's been quite wide across the centres, so that's quite a focus. It's particularly focused on the clonal crops, which are very expensive to maintain in tissue culture. Likewise, the development of cryobanking. And likewise, we've had a lot of work. I talked about subsetting, that includes figs for particular traits. And I mentioned very briefly that the work we've done on genotyping and on the GPS records of our collections have really set the foundation for what we have now, which is funding from the Bill and Melinda Gates Foundation for allele mining, approaches using environmental GWAS. So we're now using those, for example, for drought and for heat amongst a number of centres. So there's a lot of focus. There's focus also on nutritional traits. So a lot of focus on what you're saying about really, and the advantage of the allele mining again is it develops a very strong link with the breeders. There's a sort of handover, if you like, of potential or of parents with new alleles for their breeding program for key traits. So I didn't go into that a lot today. As I said, it's covered in other talks over the next couple of days. But clearly, yes, we agree that it must be conservation and use. And that includes also the use for some of these underutilized crops, which we can characterize in terms of nutrition, for example, in terms of climate resilience, but may not go through the CG breeding programs.
Yes, thank you. Another question there. Okay, you go first, Ted.
Thank you all for those excellent presentations, a great overview of what the CGIAR is doing, the new CGIAR 1. My question is around, it's been mentioned a few times, the importance of understanding what farmers are growing and why. Weighted average age of varieties in farmers' field has been mentioned a number of times. Yet the CGIAR does not regularly collect that information. Is there a new initiative? The only way you can measure weighted average age is with DNA fingerprinting data collected from farmers. Is there a plan in place for that to be done regularly so that you can monitor changes in weighted average age?
Yes, thanks. I understand there's a plan in place. I'm asking Ian to comment. Ian is just here. Ian, please comment on that one.
Thanks for the question. Yeah, so within, my name's Ian Barker, responsible as Senior Director for Strategy, Delivery and Scaling, responsible for that CDA. And that question we've adopted that question and the need to be able to collect information to do that. So as Sonia mentioned, this VarScout app, for example, is collecting adoption data, directly from the field, from extension agents. That's based on recollection data. We're also actually looking at things like AI image recognition for varietal identification and integrating that with DNA testing. So we work alongside the big DNA fingerprinting initiatives like Image, and trying to integrate that. But we have developed, if you like, a one-stop shop, if you like, an ecosystem to be able to collect all the adoption data, current, future, and past, put it all in one place. And that app, if you go and look at it, you can actually download it from the App Store. It actually calculates WAVA, and you can see it displayed on screens. But we realize we say that integrates, and we will integrate DNA fingerprinting data into that one space. But we are interested in innovating into lower cost adoption of the same data, and also collecting, if you like, proxy data. So in some cases, seed production data can aid as a proxy to identify. Not for all crops, it's highly context dependent. But we're actively working in this area and have developed at least one tool that can at least collate all the information and automatically calculate WAVA.
Thank you so much, Ian. I still have 3 minutes. I have one gentleman there, and then Professor Shimelis.
Thank you, Mr. Chairman. I want to ask about the strategy just to get, additional gene source from the area where the crops originally, just in area of the crop diverse, there is always gene involved. Just do you have any strategic plan just to get those additional genetic sources in the gene bank?
Yeah, sure. I mean, in the past few years, there have been programs of collection. So this is also based on some tools that have been developed from gap analysis, etc., which are looking at the representativeness of the collections we have. So they're addressing your issue of, not necessarily the centers of diversity as such, although it could include that, but areas which are currently underrepresented in the gene banks. So we are working on that, and over the last few years we've developed an approach which is more based on, of course this is with the national partners in those regions, and the national partners take, if you like, half the collection or whatever, and the rest comes back. So it's all done in that way, and over the last few years we've also developed approaches to trying to do that for multiple crops, so that we can be sort of more efficient in what we do. So for example, in IITA we've had collections to Sudan, to Togo, to Cameroon, but we have collected more than the IITA crops, if you like. We've collected crops relevant to other centres and also relevant to the priorities of the national programmes involved.
Go ahead,
Okay.
Professor Shimelis.
Okay. Thank you. Thanks, John. This is Shimelis Hussein from the University of KwaZulu-Natal in South Africa. I think my questions are related with the adoption and the return on investment issues. The adoption studies has been done in 2010. And there was a publication in 2014 through the DIVA project. So is there any plan to update that? Because it's a little older now, 10 years back. So if there is any initiative or any plan, strategy through the CG system. Then the other question is related with return on investment. Is there any figure, data to show some return on investment in plant breeding programs, especially in terms of revenue, the income accrued through plant breeding, innovation, variety release, and so on, and also capacity development in terms of training, postgraduate training, including postgraduate or short-term training, that can be calculated, computed in terms of really the cost that was saving the national governments in Africa by the CG and so on. So just if someone can comment.
Thanks so much, Professor Shimelis. My answer is yes, yes, yes to both. But I'll allow Ian to revisit the adoption studies in terms of updating. Ian, do you want to comment on updating on the impact studies and adoption studies?
Thank you for the question. It's a similar answer. You're right. DIVA is what, 2010, is it? Published in 2014, and has not been updated. So there are a lot of individual small-scale studies going on, DNA fingerprinting studies, household surveys, collection of seed production data. So there's a lot of data out there. The problem I think we face is bringing it all together in one place. So what DIVA did, if you like, was put it all in one place and published it at one point in time. So there is a lot of data out there, and we're working really hard to actually put all this data in one place so you can see it and to be able to calculate return on investment, based on adoption. So that we are very actively working on that. And also, again, as I say, to the previous question, trying to lower the cost of acquiring this data. I don't know how much DIVA cost, but I guess it wasn't cheap. And so that's a part of this as well, lowering the cost of collecting variety adoption data, and also, say, looking at other sources like seed production data, certification data. It can all be used to estimate adoption, but it's obviously it's a key issue for us. And if you're interested, again, look at this VAR-Scout app, if you like, on your app store, and you'll start to see that is a space where we intend to pull all this data together, including historic data, right, and going forward. So the idea would be not to do a DIVA study every 10 years. The idea would be that this would be updated every day, right, with people out in the field with smartphones actually collecting the information in the field and updating the global database every day, not waiting 10 years for the next DIVA study to be presented. Thank you.
Yeah.
And training is a very important part, and of course, in relation to market intelligence and developing investment cases. Capacity development, with our national program partners is obviously a key part of that.
Thank you so much. I think return on investment is very high both in terms of profitability and also in terms of social returns on it. My literature tells me that it's more than tenfold, but it's dependent on the crop and the impact area that we are looking at. But maybe, Michael, you may want to say something about returns on investments in breeding. Are there any returns? You can just say something. Returns on breeding. Are there any returns on breeding? Returns on breeding? Investment in breeding. Returns. How do you comment on that? No, I think I'm catching you in a way. I was asking Michael to comment on that, but I think he didn't pick the question. So I think that's my response. If anyone has a comment on it, it's more than tenfold. The return on investment in breeding is quite high, both in the private sector and in the public sector. But it's dependent on the crop and also the impact area that we are looking at. Yeah, it's not easy to measure for some of the things like return on health and nutrition, but there is. Thank you so much. I would like to close this session and thank everyone for participating here. But as you were listening from this morning, Sonia was speaking, and yesterday, in GI, we prioritize collaboration with all our partners, and we are looking at sharing capacity in all these issues that you are raising here, sharing ideas and working together. So we look forward to continuing with all these collaborations with both the NAS, the private sector, as well as university partners, the centers of excellence, etc. So we'll continue with the discussions until Thursday. All our leaders are here, Michael is here, and the senior directors are here until Thursday. So let's continue with the discussions. We'll try to summarize everything in 90 minutes, and it's not enough even for breeding alone. 90 minutes is not enough. It's a lot of work that we are doing. Thank you so much. Thank you, Michael. I think we're heading for lunch. That's what I guess. Anyone with the program, I think we're good. Okay. Oh, coming now. I was already sending people away.
So it is after midday, so good afternoon. Hello, good afternoon. Thank you so much. Unfortunately, my boss took 15 minutes away from me. I have to make it up. So welcome to this session, which is Advances in Genetic Innovations and Technology. In this session, we've got 5 speakers that are coming through. We shall try to make a bit of time so that we can go to lunch on time. So I will allow 2 presenters to come in, then we'll take questions, then the last 2, then the last one. So without taking much of your time, please help me. I think I've lost the mic. Hello? So sorry for the glitch. So allow me to call on stage Mr. Rashid Mentag from INRA, Morocco, to give his first presentation.
Good morning, everybody. So for the presentation today, I will talk on Orobanki. First of all, I would like to thank all the organizers for this nice and interesting event. And Orobanki carinata is a challenging plant for legume crops. It's challenging for me also because I had only 10 minutes to spend to answer to this challenging question. So for those who don't know Orobanki carinata, so it's very beautiful plant, but we can see right there, there is no, okay. We can see right there, and you, however, it's challenging, it's a parasitic weed for food legumes. Before that, we'll go through the importance of food legumes in Morocco, as we can say, we have about 300,000 to 400,000 hectares. And the major species, legume species, are faba bean, chickpea, lentil, and pea. And we all know the importance of the legumes crops for their nutritional, ecological, and economic importance. But these legumes are facing some abiotic and biotic stresses. The major biotic stress in Morocco, or maybe in the MENA surrounding region, we have Orobanche crenata. So it's a parasitic weed. It's holoparasitic weeds. It means that it takes 100% of its need from the host plant. And it can infest food legumes, but other crops also. And the losses to these crops can reach 100%. But this depends on the infestation level of the soil, and also depend on the legume species. So it starts with the tiny, really tiny seeds, and germinate, and then stick or attach to the root of the food legumes, and start growing and pumping all their needs, water and minerals from the roots, developing on tubercles, and so on, and there until it reaches out of the soil and produce spikes. So we can find this. Orobanche crenata is problematic in region, MENA region. We can see here shaded area, and now it's expanding to new areas. For example, the map here. It's 2007, but right now, Orobanche expansion are in the south until Ethiopia, and also some North European countries. Here in Morocco, we can see that the major, here is the distribution of the map of Orobanche crenata in Morocco, with some hotspots located in Sais region, Stat, Taza, and Tawnat. So there is the main problematic area with Orobanche. And the problem depends on the legumes species. So we can see here for pea and for lentil, for faba bean. So Orobanche crenata is a huge problem in these three crops. A little bit less in the chickpea, but the infestation depends also on the population of Orobanche. This is our recent study on the aggressiveness of Orobanche crenata populations against faba bean. So we saw that there is some highly aggressive population of Orobanche. And all over the world, or Mediterranean region, there is a lot of collaboration on Orobanche. And there is some clusters located especially in Morocco, in Tunisia, Egypt, and Spain working on this topic. There are several papers. We have around 300 papers dealing with Orobanche all over the world. And through these papers, we have many topics. For example, we have genetic diversity, we have host-parasite interaction, but 60% of these papers are dealing with control strategy. So the major control strategy used for Orobanche are cultural and physical control practices, chemical, biological, and resistance, breeding, I mean. And we can see here that the early work that have been done in 1970s using chemical control. So this is the first control method used. And since this work, there are many chemical products that have been used against Orobanche crenata. And also, in yellow, you can see the breeding programs started in 1986. And through these programs in different countries, mainly Egypt, Tunisia, and Spain, developing new cultivars that are resistant to Orobanche. For your information, there is no complete resistance to Orobanche in Fababine, for example. So the interesting thing is the use of integrated pest management of Orobanche through the use of different control methods. These control methods focus on different steps or stage of the development cycle of the Orobanche. So in our group, we focused on the introduction of sustainable pest management. So we try with microalga extract, and then this allow us to produce some interesting results and patent also. So this we can see that we did a screening on several strains of microalga. We selected 2, and then we go further with some extracts from these 2 strains. And we can see here the reduction in terms of infestation in Fababine. And also, we noticed also that there is a biostimulant effect of this extract. So we go deeper on the understanding of how it acts. So we did some metabolomic analysis, and I go fast and say that there is, for example, for Fababine, there is a synthesis of alpha-linolenic acid that could act as a precursor for a synthesis of jasmonic acid. Also, we try to use other sustainable control methods, as we know that glyphosate, maybe it's uncertain that that is highly used in control of Orobanche. We are not certain that this product will be available in the coming years. So we try to develop other sustainable control methods. So we use arbuscular mycorrhizal fungi, and we see here there is some strain reducing infestation in 2 different cultivars. And we can see that some interesting glomus strain are really effective for controlling Orobanche in Fababine. And also we try use Rhizobium strain. We can see here significant reduction of the Orobanche. In the middle, we can see with Rhizobium, in presence of Orobanche, we have small Orobanche infestation event compared to the control where there is a lot of infestation, Orobanche infestation level. Also, with sustainable control methods, we try intercropping using coriander and fenugreek. And these methods, it's under control of 3 factors. It's three parameters, I mean. There is the legume species. It depends on legume species used, and also it depends on the intercropping crop used, coriander or fenugreek, and also it depends on the infestation level of the soil. As we can see for pea, for example, intercropping with fenugreek, no difference with pea alone compared to in presence of coriander when we have high reduction of the infestation. So, in conclusion, we can say that Orobanki is a serious and expanding problem in many countries. Several control methods have been developed and effective against mainly chemical control and also breeding programs that have developed a lot of cultivar resistant to Orobanki. And the use of integrated pest management is the best way to overcome this problem. And it's really interesting to enhance our knowledge on the interaction on legumes and Orobanki to have a clear idea and to develop. And this will be used or will be targeted through the omics technology. But the last one is we need an effective technological transfer to the farmers. Here in Morocco, for example, we have some interesting control strategy, but these control strategies are only in laboratory. I mean, we should use them, transfer them to the farmers. At the end, I would like to thank these nice people for contributing to our studies on Orobanki and legumes interaction. For your information, among the 10 top active researchers in Orobanki studies, we are 4 scientists from Morocco. So it's interesting, and thank you very much.
Well done, well done. Thank you very much. In the interest of time, please could you have me, Lamie Gauchi, to come and present to us. So the title of your talk is, Does the variety have any effect on productivity and quality in mixed cropping systems of cereal and legumes, case for durum wheat and faba bean?
Excuse me. Ladies and gentlemen, it's a real pleasure to be among you today and to make this talk just after the very nice talk of my colleague Rashid Mentag, who's here to defend also the legumes. And we are here both together to defend these very, very interesting crops. So actually, I would like to start by saying that as a problematic, and as we all know, we all know that we are facing challenges where we have to change our paradigm of agriculture, because we are aware today that we have limited resources, but unlimited scientific advance. And this is what can help us actually to install let's say, a sustainable agriculture. So what we want is that we want to keep the productivity going high, but we want the vulnerability of the systems to go down. And for that, there is just one key factor, that is the diversity. I just need to go, yeah. So the biodiversity with its levels, whether on the interspecific or intraspecific level, these are actually the factor, this is the factor that is going to help us to actually go toward the sustainable agriculture. So legumes, of course, are offering a lot of assets, whether on the I mean, the assets for the environmental and health level, or also, I mean, the ecosystem service crop, or as a human and animal nutrition. I'm not going to go the diverse advantages of having legumes, whether in the soil, whether in our cropping system, but also for our nutrition. But still with that, when we go and we see the cropping system, we still see that cereals are actually occupying worldwide more than 42%, whether legumes are just occupying just 6%. So which is, of course, providing a great unbalance between those two crops that should be a little bit balanced to their acreage and their occupation. In Morocco, it's also the same. And of course, we do have here cereals that are 59% within the acreage, and only 3% are dedicated to legumes. And they are mostly in Gharb, Shawiya, Rif, and Sais, the regions where we are cropping legumes. And in these legumes, faba bean, as it was said, with other legumes, are the one that is most important and most ancient also in our Mediterranean system. Now, if we want to use legumes, we have different ways. And among these ways and practices, we do have intercropping, and we have also rotations. So if we see intercropping, which is a very interesting practice that is very, very much appropriate for the system and the agriculture we want to develop, the future, the present and future agriculture, we could see that intercropping and using legumes is, of course, one of key options for these agronomical practices. It could be used just in varietal association. We could have association with perennial crops and also in species association. What it can do, of course, it is increasing yield, it is increasing quality, it is also controlling weeds and diseases. Now, most of studies are addressing the interspecific diversity, meaning that we are looking which cereal with which legume is going to give the best combination if we are to address yield, quality, disease, and weeds. But, I mean, if we take a given cereal and we take a given legume, the question here is to address which variety, which genotype in a given cereal and which genotype or variety in a given legume could give the best combination to provide a good yield, quality, and to control disease and weeds. And for that, so we have very few studies that are addressing the intraspecific diversity and to see which genotypes are actually the most dedicated to intercropping if we have to apply intercropping. So we have been digging for some studies, and there are very few, but most of them are showing that there is an effect on yield, there is an effect on flowering time, there is an effect mostly on yield and flowering time, and these are some intercropping, studies where we address the genotypes, the effect of the genotypes on the intercropping, and we have like 25 until 80% of the variation of the trait could be affected to the genotypic variation, and this is very important. In our study, we have taken durum wheat, as it is a rustic crop in cereals and very, very adapted to the agroecological conditions in Morocco, and faba bean, as it is the most important legume. And we have these research questions. Our question is first to say, is there an influence of the varieties on the performance of the associations? That's first. Second, is there a specific profile for the varieties that are best performing in association? Third, what are the trait correlations between the soil cropping and the cropping system, in association? Do we have a trait correlation for both? And, of course, should we develop specific breeding programs that are to be developed for the varieties addressed to the intercropping systems? Because that's the question. Are we going to have just a classical breeding program and then we could use it for intercropping, or should we have specific breeding programs for that? And for that, I mean, for this question, we have addressed them in an ARIMNet project, which is D4DECLIC, with a consortia of researchers that I would like to thank from here. And this task was addressed for Morocco, and we have been testing that in 2 locations, in Tifelt and Meknes. We have taken 10 varieties of durum wheat and 10 varieties of faba bean based on their 1,000 grain weight, precocity, and also height. And we wanted to exhibit the interaction between the different varietal profiles. This is why we have taken very different genotypes. So, meaning that we have been ending up with 100 combinations. That was not a very easy work, but it was a very challenging one. Those 100 combinations were sown in a replacement design, and where we have developed 140 plots actually that were used in 2 replicates and in those 2 locations and for those 2 years. I'm not going through the experimental design, but for those who are interested, it's an alpha lattice design, and with, of course, partial blocks of 7 plots per block. Well, that's actually just one of the trials we have been installing, and these are the partial blocks. We have been taking several traits, of course, traits of growth, of NPK analysis, of yield and yield components. These are traits we have taken at the plot level and others that we have taken at the plant level. And we have been doing, of course, several analyses going from the ANOVA until the multivariable analysis. As for some results, because I'm not going to present all of them, I have just a few time for that. So first, that is very important for us is not to see whether the 2 cropping systems are different. Of course they are. But what is very important is to see the interaction, whether there is an interaction between the system, the cropping system, and the genotypes. And it was different for wheat. So it means that the genotypes of wheat were acting and behaving differently whether they were in the sole cropping system or in the mixed cropping system. For faba bean, it was not the case. So it means that wheat was much more sensitive to faba bean, to the mixed cropping system. We have been also addressing the LEAR, which is the land equivalent ratio, and where we have seen that genotypes in wheat and also genotypes for the combination did have a very different land equivalent ratio. Through the analysis, we had like 25% of the combinations did have a gain of yield that were amounting up to 80%, 18% of the grain yield. I mean, it was done for 25% of the combinations, and this is, of course, of interest. And when we have done a principal component analysis to understand, because here the objective was to understand what profile of the genotypes are the ones that are more adequate to the mixed cropping system, we have been seeing actually that when you see the analysis for durum wheat, you see on the right side, so the group on the right side, these are the ones that are behaving very well on the mixed cropping system. And the other ones on blue, they are poorly behaving on the mixed cropping system. And we have seen that earliness is the major factor that is describing the varieties that are well behaving in the mixed cropping system, whether we take it in wheat or also in faba bean. Earliness is a key factor for the genotypes that are to be used for the mixed cropping system. As to some conclusions and recommendation, I just would like to emphasize that there is a significant difference between the species and cropping systems for all parameters, that we have it also for the specific analysis for both species, and then that for wheat the interaction was more significant for the growth rates and production trait, but for faba bean the interaction was significant just for the flowering trait. So mostly it is on wheat that we have to be focusing. On yield, we had like 14% of gain for wheat and stability in grain yield for faba bean. We have up to 18% or 18 quintal per hectare, deciton per hectare for, as a best performing combination. And there are some genotypes who are already interesting for this combination that we have been identified. And also we have noted an increase of the protein rate of 1.61% and an increase of vitreosity of 15%, which is also very important. So, to be sharp, because I started with questions, I would like to end with questions, although I ask you to be very indulgent because I wrote some answers that have to be taken carefully, and we are still going to validate them. Is there an influence of the varieties on the performance of the associations? Yes. Is there a specific profile for the varieties that are best performing in association? Yes, earliness in flowering and heading. What are the trait correlation between the sole cropping system and the cropping system in association? Mostly yield, yes, it is correlated, which is a good news. It means if you are doing a good job for sole cropping system, you are doing a good job also for the mixed system. Should we have specific breeding program? Not necessarily at the beginning, but we may have it at the end, because we could have some genotypes that are where we could use the interaction at the end, I mean at the late phases of the breeding program. With that, I would like to thank the funders, the consortium, the professors that were part of this project, the technical assistants, and also the student, and I would like to thank you for your attention.
Well, thank you very much for those interesting talks. I will invite some questions for the presenters. Do we have the roving mics?
Thank you very much for the nice talks. Now I'm really wondering why the devil is always beautiful. I'm thinking of Orobanki and Striga at the same time, and I'm rethinking color purple and pink. But my question is, since the legumes are struggling with Orobanki and the grains are struggling with Striga, are there any cross-learnings that can happen between the two teams that are working on Striga or Orobanki?
Okay, so for Orobanki, it's a little bit different compared to Striga because it's holoparasitic weeds. It means that it can never grow if there is no attachment. For Striga, we have a little bit some autonomy of the plant until it sticks to the for example to the maize. So, but there is an interesting work that have been done in US with a consortium of US scientists working on different kind of parasitic weed. It means from, if we go down from Orobanki, that depends 100% on the until Striga, and also Triphysaria, which an interesting model also for parasitic plant. And they did some transcriptomic work, and they find that there is some genes that are responsible of such interaction between host plants and parasitic weeds. So, I mean, the approach is similar. It's parasitic weed, but each one, even if we take Orobanki with Fababin, are not the interaction is not similar like Orobanki Cernuia with sunflower. So for the last one is gene for gene. So we have races in this interaction. But for Fababin, for example, there is multigenic interaction. So it depends on its case by case.
Thank you to both presenters. My question goes to the second presenter. Part of the trait that you highlighted was earliness. So intercropping is one of the major system of production in West Africa. So we are very much familiar with that. Now, we mostly intercrop legumes and cereal, but the timing of planting is not in order. So most of the time, you have the cereals coming first, then the legumes coming afterward. So when you say earliness is a focus, did you factor in the time of planting, or they were all planted at the same time? Thank you.
Thank you for your question. Actually, what we have done, we have tried a little bit to see for our conditions in Morocco, and usually for our conditions, faba bean and wheat, they are planted at the same time. So we have been planting them at the same time first, and then we were planting at the half of the density, because what we wanted is to have, to exhibit mostly the interactions. So that was the first, the experimental approach, knowing that we are not very much familiar, less than you, in your context with the intercropping.
Yeah.
Thank you very much, Chair. I want to congratulate our two speakers, Rashid and Lamia. Very nice talks. I have a question for Lamia. This is interesting work. My question is about durum wheat varieties. Probably you don't have enough time to list the varieties that has been used in your study. I'd prefer have names of varieties that you used, if you remember of the name of some of the varieties. Do you have old and medium age and recently released varieties in your study? That's my first question. The second question is about, yes, I will say earliness is very important. Probably, I was talking with my friend Liban, it's a question of early vigor and also the germination for a variety, if it has an early germination that can be established very early, so will allow take advantage of the resources that probably have a good yield at the end. And I agree, we could not have special breeding programs. No-till are asking for a special breeding program. If we want to add also intercropping breeding programs, we don't have enough human resources and financial resources for that. And thank you so much for your talks, both of you. Thank you.
Thank you very much. Actually, you are right. So for the varieties, what we have done, we have taken all the varieties that are used in the market, because this is in the Moroccan market. And they are not just middle-aged, but all the varieties. We wanted to be a little bit, not to do just something experimental, but really to see within the varieties that are used, the old ones and also the new ones that are used in the, in the cropping systems in Morocco. So that's the first answer. And I have presented the list, but it was just a little bit too fast to see it, but I could share it with you. The other question that is very interesting is that you say we cannot make specific breeding program. Well, the good news for you is that the scientific results are just doing you a good favor in not pushing you toward that. But if we would have found that actually there is no correlation between yield in sole cropping system and mixed cropping system, then you would have to develop it if intercropping would be, an important cropping, practice in our system. But we did find that there is a correlation, and this is a good news for that. Still, I want to insist on the fact that not at the beginning, but there is an interaction, and this interaction is to be taken mainly at the end of the programs, meaning that you could do screening when you still have a lot of genotypes in your breeding programs, at some stage, to when they are already developed, but not to take the best variety for the sole cropping system to make it in the mixed cropping system, but still to use it in a little bit some stage earlier to take from a very large number of genotypes and to screen them for the intercropping.
Thank you very much. So we'll take a question there, one more here, then we'll roll it over when we finish. But before you ask, I want the next two presenters to come closer, so we cut on time.
Okay, thank you, Chair. And my question goes to Faba Bean, because in the conclusion there is a very clear control, measures which are there. But he said it's only in the lab. So how we can bring that to the farmers? Are they did really, the extension on that? And the bundled technology is really easy for farmers to take it up. What is the next? I mean, you have the technology, then should it sit there?
Yes, I think that the major problem is technological transfer to the farmer, that's it. So we have, for example, for the use of glyphosate, it works. Depending on the impact of environmental, so all we know that there is some environmental impact of such chemical products on the environment and the health of humankind. So, but it's still efficient if well used, and maybe in the large farm, it's used and it's efficient. The problem is small farmers. They don't have information about how to use the product. Sometimes to prepare, we need a lot of water, and logistics, it's not easy to deal with. But I think the major, we have to emphasize on the how the farmers, small farmers, get this information, and extensions are invited to use such technology and to transfer them to the farmers. I think this is the major challenge at least in Morocco, but even other countries. Yeah, I have one question for Dr. Louti. So,
is it allowed?
No, it's allowed. It's allowed. It's interesting for mixed cropping and intercropping, but how about the competitiveness? Did you notice that some competitiveness among legumes and cereals?
Well, of course, all the relation, and I have spoken a lot about advantages. I have to say that I was not so fair with the intercropping because I was just citing what is positive. But what is negative is that, of course, you have to deal and you have to manage the competition. And this is why maybe other studies should focus on the sowing densities. Maybe the one that were used are not the proper one. The second thing is, as you said, you could have indexes of competition to analyze. And faba bean, of course, was much more suffering than wheat, which is completely, this is why the yield of faba bean is, at the best, it is stabilized. Otherwise, it is going toward a loss. But for wheat, of course, it is an advantage to have, because you have to mind, a wheat plant is better with a faba bean plant next to it than to another wheat plant for a competition. So this is why, from the competition point of view, faba bean is suffering much more. But what we are doing here is seeing a little bit whether the competition, the positive effect on wheat, will compensate the competition on faba bean. And this is why, beyond what we have been studying, we have to see a little bit what is the optimum, agronomical practice that would lead us to a better yield, maybe for both, and to have a compensation between both species. Thanks. Thank you a lot.
Sir, 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 please.
There is another question there.
Hello, everyone. I'm Mohammed, a PhD student at INRA Morocco, and I have a question for Mr. Rashid. My question about Bron Prep, as you have indicated, several methods exist to control orobanki, such as chemical methods by using a chemical solution, but exist different other methods such as control of strigolactone, but this control have many secondary effects on plant growth and development. Donc, so how controlling development of strigolactone and controlling the healthy growth and development. And thank you.
Okay, thank you. We all know that strigolactone is the chemicals product in the exudate of the root at food legume root level, and it stimulates germination of orobanki. Maybe 20 years ago, in Netherlands, they developed synthetic strigolactone to allow germination of seeds. But it's difficult to use this strigolactone as to decrease the orobanki stocks within the soil. So it's very expensive, just products. We use the GR24. It's for 10 milligrams. It's cost 400 or 500 euro. So it's very expensive. But there is some elicitors that could be used, others elicitors that could be used efficiently, and we have a recent paper on the use of NEA to overcome this process. We have nice results with controlling orobanki in faba bean and in lentil. We know now there is no exact relation between the decrease of synthetising strigolactone and resistance. We have, for example, two sensitive varieties. When we do some analysis on strigolactone, we can find that one is releasing small amount of strigolactone, the other is huge amount. So it's not a correlation between the production of strigolactone and sensitivity to orobanki.
Allow me to pay you, the two of you, during lunch. I think this is stimulating, and I must say, let's clap hands for these brilliant presentations from the team from Morocco. Thank you so much. So our next speaker is talking about sparse testing and sparse phenotyping from CIMIT Mexico, Mr. Yuen. Let's clap hands for him. Did I say the name right?
Yeah.
Okay. We'll try to have about 10 minutes.
Okay. With this?
Yeah, that's good.
Okay.
Yeah.
Good afternoon, everyone. We talk about sparse phenotyping and sparse testing. The idea of this kind of phenotyping is use the knowledge we have on genome selection and predictive models and the use of genotypic data, molecular data, to do prediction, and we can use this kind of information to try to improve the breeding program, especially at the early stage. The idea of sparse phenotyping and sparse testing is not to test all the lines, I will talk on maize, all the lines with all the tester or all in all the environments. That is the main idea, to save some resources and use those resources to increase the population size or the sampling of the target environment. The data we used to do this work was was developed in the maize program in Eastern Africa, in Kenya, and for stage 2, during 2 different years, we work in 2 heterotic groups. We have then 4 different sets of data, and the location you can see there, Kiboko, Kirinyaga, Embu, Kaguru, and Kakamega. In 2018, we have 1 less location. We work with around 400 genotypes, plus some checks, different number of checks in each year, and we use RAM markers, random amplification markers, in a number of around 4,000. The analysis of the data, what we started with the complete test of the genotypes. We have the 5 location, 2 replicates in each location with an alpha lattice, and the idea is to remove some genotype from the original data set, the complete data set, to generate different strategies or scenarios of sparse testing and sparse phenotyping. We use, well, that's okay, the data. And this is the design that we use. In the rows, you can see the 4 different sparse phenotyping designs, and in the columns are the sparse testing designs. Yeah? How do you interpret this? In the first, you have the environment, E1 to E5, yes, and all the genotypes are in 1 set, and all the genotypes are tested in the 5 environments. In the second row, we have 11 sets of genotype. We split the complete data set of genotype in 11 different groups. The first set is tested in all environments, and the remaining ones are tested only in 3 environments. In the second, in the third row, the second option, we have also 11 sets of genotype. The first tested in all environment, and the other set only in 2 environments. And in the last, the other sets are tested only in 1 environment. For sparse testing, we did more or less similar, the different columns. The first case is the complete testing, in which we have all the genotype, only 1 set in all the testers. And in the other, we have, we use 2 testers for some sets, then 1 tester per set of genotype, again 1 tester, then 2 testers, yes, and at the end only 1 tester. If you see the sparse testing 3 and 4 are the same, yes, we have some sets of genotypes only with 1 tester, but the difference between both is that is something that we need to think when we plan this kind of design, is how many genotypes we put in each set. Yep. In the option 4, we put more genotype in sets 2, 3, and 4 than in set 1. And then in the option 4, we are reducing the total number of plots in 60%, around 60%. In the option 3, we reduce the total number of plots for phenotyping in around 30%. That is the difference between 3 and 4. And in all the cases, we try to maintain more or less the same number of plots that we save in order to compare more the designs. Then at the end, we have all the combinations, the 4 different sparse designs, plus the 6 different sparse testing. After we develop the different scenarios, we evaluate the results comparing the breeding values of the using sparse testing, sparse phenotyping, with the breeding values of the complete data analysis, using two statistics, the Kendall coefficient correlation and comparing with the mean of the selected genotypes under two different conditions, selecting 20% or 10% of the best genotypes. Then in this figure, you can see the Kendall correlation. Each quadrant is for the 4 different sparse phenotyping. 0 is no sparse phenotyping on the top left. The others are different sparse phenotyping. And each box plot corresponds to the different sparse testing design. That is how many testers we use for the different lines. I'm presenting results only for the heterotic group A in 2018, but the results or the pattern of the results are the same for the other years and for the other heterotic group. You can see that correlation goes more or less around 0.4 to 0.8. Of course, when we have more sparsity in the design, we have a low precision, low accuracy, and maybe it's interesting to compare the case 3 and 4, in which we have the same sparse design, testing design, but different percentage of saving plots. Yes, the red and the green, these two options, the 3 and 4. Yeah, and then we can see that the case 4 is similar to the case 6. Yes, that means that we have more or less the same accuracy using a lower number of testers but more genotypes, or using a lower number of genotypes with more testers. If we consider the mean of the selected genotypes using 10% of the top genotypes, I think this is maybe more interpretable because we are talking about tons per hectare. Yes, the red line is the means of the genotype selected with the complete data set. Is 7.86 tons per hectare. And of course, in the other cases, we have the box plot for each case of the simulation. We can see also the same pattern. More sparsity, we have more difference with the complete data set. The important thing here for me is that we can say, okay, we have more or less accuracy, something that we can expect because we have a lower number of replicates. But the point is how much we are losing for using sparse phenotyping or sparse testing. Yes, this is under 10%. Under 20%, we have a similar pattern. Yes, of course, the means are a little lower. And in the last figure, we have the number. Yeah? And what we can see, in yellow are highlighted the cases in which we don't have a sparse phenotyping, but we can see that we lost around 8 kilograms per hectare, yes, 273, 150 in the last cases. It depends, of course, of the year, depend on percentage of genotypes we select. What we have to consider to evaluate this number is the genetic gain is more or less 100 kilograms per year. And then if you see in the worst cases, we are losing all that we can gain in 1 year of selection. But the main idea is that, as a conclusion, yes, first is that the use of the genomic selection allow us to do this kind of sparse evaluation, phenotyping and testing. Then we can save some funds. We don't need to do so many crosses. But the important thing is that to recover part of the loss in precision, we need to do something different. And the main idea is that if we reduce the total number of plots in 30, 40%, then we can increase the population size, and then we can get a better genetic gain. Or we can test more environment, and then we can get a better sampling of the target environment. Both things will improve the genetic gain. I didn't present the result, but we found similar results using the pedigree. Then we can avoid to use genetic molecular data, And also we found more or less similar results on wheat. And finally, the point here is that the breeders have these numbers in order to decide how to improve the breeding program. Of course, an important thing is to consider the cost and the logistics of using a complete testing or a sparse testing. This is part of the literature in which you can read some results and in which we base the analysis. And I want to thank the donors and people from the Göttingen University which work in this project. Thank you.
Thank you very much. Would you like to invite our next presenter, whose title is Development and Deployment of Climate Resilient Wheat Varieties for Food to Ensure Food Security in Africa, Mr. Wuleto Tadis from Aikada, Morocco.
How is it working?
Yeah, it's just
on the green.
On the green.
Okay.
Yeah, I think you are.
All right. Thank you, Chair. I'll be talking on development and deployment of climate resilient wheat varieties to ensure food security in Africa. I don't know how many of you attend. The mic is not working. How about now? All right. So I'm not sure how many of you have attended the Wheat for Africa workshop in 2012 in Addis Ababa. And I would like to show you in the coming slides what happened since then. Is really Wheat for Africa become a reality or not? So the team who are working with me is indicated in this slide. It indicates different specialists from seed science, pathology, entomology, also quality genomics. So for further information, I have put also my contacts there. Okay, an important slide here. If you see the wheat production area from 1961 up to 2022, it remains the same, around 220 million hectares. But the production has tripled from 220 million tons to 775 million tons. What happened? The development of the semi-dwarf wheat varieties, which are input responsive, has helped significantly for this. But also, the availability of inputs like fertilizers, irrigation, machineries has played significantly. And not only this, but the favorable policy scenarios, availability of good policy environments has played significantly. Imagine some of the great breeders who has contributed for this, like Dr. Borlog and my mentor Rajaram and many others, Dr. Osman Abdullah, and some of you who are sitting here. Each one of the wheat breeders have contributed significantly for this huge level of wheat production globally. When it comes to Africa, Africa grows wheat in 10 million hectares and produces around 25 million tons, but consumes 75 million tons every year and imports 55 million tons. and has to pay, it's not free, $20 billion every year. So can Africa grow wheat and feed itself? So the challenges, both biotic and abiotic constraints, social issues, conflicts, infrastructures, and sometimes even if you have the money, you may not have the way to buy. But are there opportunities for Africa? Yes, there are big opportunities to grow wheat both vertically and horizontally. There is a young, productive workforce. There is a huge market. And also, there is a possibility to make agro-processing industries, job creation possible in Africa by growing wheat. But there are key challenges which we need to focus as breeders, the biotic and abiotic constraints. And from these, the CADA breeding program focuses on rustes, septoria from the biotic constraints, and from the abiotic, the major ones are heat and drought. As you can see here, in the first slide, yellow rust, when it happens in the savanna, causes significant damage in Ethiopia and the rest of the savanna and Morocco. And also in Morocco, drought, when it happens, it causes significant damage, up to 100% yield loss. So focusing on these key constraints, our breeding objective is to develop a germplasm which is high-yielding, stable, and adapted to the region with resistance to the key biotic and abiotic constraints. And of course, we develop also germplasms for pre-breeding purposes, gene identification, marker identification, capacity development, and scaling of the wheat technologies to farmers, along with our key partners, the national programs. So in doing this, identifying a key methodology, strategy is very important. We used to have double haploid breeding program, modified shuttle breeding program, and inter-country shuttle breeding program. And we have taken analysis of this, and the number of years for a double haploid takes 5 years from crossing to distribution of international nurseries. For modified shuttle breeding, which we are using currently, that takes only 4 years, but the inter-country shuttle takes, on average, 5 years. So if I focus on the modified shuttle breeding scheme, to increase the genetic gain, of course, we need to increase the selection intensity, the heritability, the additive genetic variants in a short period of time. Important is characterizing and identifying key parents. That is where it matters. And of course, the key locations, the machineries, the availability of skilled breeders, they do also play in this, not only shortening the time. Most of the time, we just focus on shortening the time. In shortening the time using the modified shuttle scheme, we have 2 cycles in the greenhouse, 2 cycles in main season and off-season in the field, and we advance the generations. The stage 1 and the stage 2 trials are carried out during the main season without any shuttle activities. We do implement marker assets selection and key location testing of the stage 1 and the stage 2 materials. The key locations we follow are Egypt at seeds station for measuring yield potential, Morocco for drought tolerance, Ethiopia for disease resistance, the Kulumsa station, and of course, Tarbul, Lebanon station for cold measuring and also for wide adaptation. So with this scheme, for the last 5 years we adopted, it was able, we were able to reduce the number of years from 8 to 10, which we used before, in 4-year cycle. From crossing to distribution of elite germplasm to the national programs, it takes only 4 years. So in addition to the key locations I already mentioned, we have also a key regional REST lab, which I bring it here for your information, that this lab is a very outstanding lab, modern lab, built by CIMIT, ICARDA, and the Turkey National Program. We need to utilize this as key phenotyping platform for arrestees. We do employ marker asset selection using the selected functional markers for the traits I indicated here, and we have developed germplasms that combines yellow rust, stem rust, septoria, and leaf rust. Following the key location shuttle breeding, we characterize the germplasm for heat, drought, rust, and yield potential, and we identify germplasms which do combine traits, like this one. The ones on the right corner side, they are high-yielding, drought-tolerant, and heat-tolerant, with disease resistance as evaluated from the columns allocation. This group of germplasms are elite germplasms, and these are the ones which are going to be evaluated for quality traits and distributed to partners through international nurseries. For bread wheat, the protein quality is very important. Flexibility and also the stability. These are very important traits, and we do measure this before we distribute germplasm to partners. And we have also tested the zinc and iron and selenium contents of these elite germplasms. We didn't breed intentionally, but some of the germplasms, they do possess good quality of these micronutrients. So germplasm distribution from ICARDA focuses basically for Sewana region and Sub-Saharan Africa region, but we do also distribute upon request to the rest of the world, Australia, Canada, Europe. But special nurseries, we compose and distribute to the key partners, and most of the variety release comes from these nurseries. And in the last 10 years, from this germplasm, more than 70 wheat varieties have been released directly by the national programs in Sub-Saharan Africa and the Sewana regions. So we do also have started a hybrid wheat breeding program. We use first the chemical hybridizing agent, and then recently we use the genetic system, the Blue Allurine system, in partnership with Sydney University, and this is currently being carried out in Ethiopia, Bangladesh, and Pakistan. Capacity building is another component. We do train students in different areas, rust, yield potential, quality, drought, and heat, and of course also in short-term training. Another important activity is outscaling. It's not the number of varieties that are registered, but also like we publish in QTLs, it will be just of nothing, but important is to partner with the national programs in big projects like the TAT, the SAD ATC project, and to promote these technologies. In Ethiopia, Sudan, Nigeria, and other 12 sub-Saharan African countries, we have done this for the last 7 years, and significant impact has been established, especially in Ethiopia. The wheat production area jumped from 1.8 million hectare to 3 million hectare, 1 million hectare under irrigated, and this is basically carried out by the national program and the Ethiopian government. Policymakers do play key role in such achievement. In Ethiopia, according to the government report, they achieved from 4.5 million tons wheat production 5 years ago to 15 million tons of wheat production in the 2020 fiscal year. That is very significant, huge amount. And can we repeat this in other countries? In Sudan, it has been significant reported in Nigeria and also in other African countries. The methodology we followed, innovation platform, clustering of farmers, and also credit arrangement for farmers, the involvement of policy makers and development banks, they do all play a significant role. So in summary, it is important to increase genetic gain and also develop varieties combining major and minor genes following the key location approach, lowering the yield gap, increase the rate of variety turnover, public-private partnership, development of hybrid wheat variety is a potential in the region we see it, and regional collaboration and investment in wheat technologies and development, production, and marketing are very important issues. Thank you, Chairman.
Thank you. Some question time? We'll take a few questions. Yes, sir.
I would like to thank both presenters for the nice presentation. Maybe my question goes to Dr. Uleta. I think just the case of wheat, now just wheat rust is the major challenge for most of wheat growing areas, and most of the varieties released, just as far as I know, have few genes of resistance. Maybe just what needs to be done to have a durable wheat rust resistant varieties. Is there any strategy we are currently used to do that? And the other question I have for you is just now the question of estimating genetic gain is now in the shelf. So just there is an advice, just we normally practicing just do field evaluation without spraying chemicals. The question of spraying chemicals to estimate genetic gain is just arising. What is your advice to do chemical screening, spraying chemical for wheat rust just to estimate genetic gain in wheat breeding?
Okay, thank you, Dr. Taye. Regarding the problem of rust durability, that is an important point. Especially the national programs have faced many challenges. You have seen many varieties have been released, even adopted. But after 3 years or 4 years, they are wiped out again by viruses. The strategy for that purpose has been advocated adult plant resistance, APR. But the issue with the APR is it depends on the amount of inoculum during the time of screening and also the season. So during those seasons, you may have seen that an APR type of reaction and the variety is released, but then in East Africa, the rate of mutation of the pathogen is very high. So my suggestion is, that is what I presented, breeding using combination of major genes in a minor gene background. That would give durability. And advise farmers to apply a general fungicide at least one rate minimum, because that is also the experience in the rest of the world. That will decrease the rate of inoculum buildup in the country, in the location where it is growing. Second question, genetic gain measurement. We have published, there are different protocols. So long as we want to measure the yield gain, we need to spray. Spray, protect, and then see the difference between varieties that you measure in terms of quintals per hectare. But it is important to put them at least in 2-3 years at contrasting locations. Then you can see the difference very clearly. But it has to be protected. Yield protection is very important.
Thank you, Chairman. Thank you to the both presenters. I have a question on sparse testing and sparse phenotyping. So when we look at genomic selection, are we saying, maybe if you recommend, is it a substitute? If I don't have the resources for genomic selection, can I use sparse testing, sparse phenotyping to make an informed decision to reduce such cost of phenotyping or fast track my selection process? That is one. The second question, when you look at the sparse testing and fast phenotyping that you did, you already have, let's say, a training population. You already have a set of data that you use. Now, if I don't have that data, I have the population at hand. Is there a system that I can put it in a computer and simulate and run the simulation, and then I have a decision? That is your question. To the second presenter, can I? Can I ask the second presenter?
Let him answer so that, yeah,
All
there's
right.
no information overload.
Yeah. This is one, what I present is a case of genome selection. We don't reduce the time, no? Because we need to do phenotyping, and we are using one cycle for that. That is the first thing. The second thing is that we need to think all those new methods or methodologies that we can use on breeding have to be evaluated in the context of the complete breeding program. Yes? It's not just to say, okay, we'll use this to reduce the cost. Yeah? The idea of sparse phenotyping, sparse testing, is to use the knowledge we have on genome selection to improve the breeding program. How we improve it? Reducing the cost of phenotyping, of course, and not the cost of the breeding program. We use the funds we save for phenotyping to increase population size, and then we will increase the genetic gain, or we will increase the intensity of selection. The same, we can test more environments, and then we can have a better evaluation of the target population environment. That is an important thing. It's not just to use this to reduce cost. Given a fixed cost or a fixed budget, maybe, a fixed budget, we need to decide how to allocate. We could put money on phenotyping, or we can put the money on genotyping, or in increase the population size, but it depends on other kind of things that you as breeders know much more than me. You need to do the crosses, it has a cost, you have to maintain the lines, yes, you have to increase the seed, the amount of seed, there are other many things. The second point about the training population, for sparse phenotyping, sparse testing is not a big issue because you have a close population in which is your training and you are predicting. And in all the cases, all genotypes are tested. Not all the test crosses, but all the genotypes. Then the relationship between what we are predicting and what we are using as a training works well in that specific case. Of course, if we have historical data, we can try to use that historical data to improve the training population, but I guess it's not necessary in applying sparse phenotyping.
Okay, thank you. You had another question?
Yes.
Okay, you can
find. To the next presenter, normally producing hybrids for self-pollinating species can be a big deal. So I was really interested when you showed the slide on double haploid, and then in part of your presentation you talk about the use of chemical hybridization agent. So moving forward, if you are going to support the NARS, that's the National Agricultural Systems, are you going to recommend them to use CHAs in developing hybrids, or it's the same DH or conventional hand emasculation? Thank you.
No, there wouldn't be hand emasculation for self-pollinated crops. So what, there is the hybrid wheat production in Europe using the chemical agent, right? So more than 500,000 hectares of hybrid wheat is currently grown there. So it is an existing practice. But what we are saying now, what we are trying is a genetic factor that enables to the development of a sterile female. Then you have a sterile female using your fertile male, grow in two contrasting rows, and then you harvest the F1 seed and give the F1 seed for farmers, like hybrid maize. That is the system we are developing. It is very easy and cheap practice.
One last question.
Yeah, just following actually the question that was put, I would like to have your opinion as to the hybrid development. Is it really worthy to do it on wheat, considering, of course, that it's an autogamous crop, so it means that the heterosis is not that much interesting as it is for the other cereals that are partially allogamous or allogamous completely. So of course there is a gain, but this gain is this gain really compensating all what you have been citing, the development of the mother, of the father, because you have to have the maintainer, the sterile line, etc., etc. So is it really, are you going in favor of that? And is it really compensating the costs of the breeding, the exploitation of the heterosis, is it really compensating the costs of the breeding? Thanks.
Very good question. So the farmers which grow hybrid wheat, as I said, in China and also in Europe, they are benefiting because they have carried out the cost-benefit analysis. As I said, in Europe now, there is 500,000 hectares under hybrid wheat. So what we are doing, it's an experimentation still with Sydney University, is a genetic system that wouldn't go through what we call the 3 parent systems. It doesn't go that way. So it involves only 2 parent systems if it's established. And then about heterosis, we have tested heterosis level, and we have reported about 27 to 30% heterosis. So for self-pollinated crops, as you know, the seed cost production is the one that discourages. Otherwise, even at 15% heterosis, it would be productive. But if we develop a strategy like this one that reduces the cost of seed production, why not? So, but this is under trial, but using the chemical agent, as I said, there is already a system that has been implemented in Europe.
Okay. Well, thank you very much to our presenters for now. I think I can excuse you for now. I'll take this moment to, let's clap hands for them. Wonderful presentations. Thank you. So for our last presentation, it's coming from Henriquez Calaio from IIM in Mozambique, speaking about cowpeas. Henriquez? He's not here. Okay. I've just been informed that Henriquez is not around, so that brings me to the end of this session, and remains for me to thank you very much for participating and attention, and to our presenters, thank you so much for the work that you put in the presentation. I think we are suitably informed. With that, we come to the end of this session.